vanguardia de la investigación científica que se realiza en el mundo. La Fundación Ramón Areces, institución de ADN científico donde las haya, procura estar alerta sobre los avances en la medicina y en la investigación médica, sobre los nuevos tratamientos y tecnologías que están revolucionando el cuidado de la salud. En el simposio de hoy, profesores líderes del MIT hablarán sobre medicina de precisión, inmunoterapia, terapia genética, nanomedicina o sobre inteligencia artificial para mejorar el diagnóstico, el tratamiento y los resultados de los pacientes. Recuperamos en esta edición la intervención de representantes de startups, a las que también doy mi más calurosa bienvenida, y en el día de hoy creo que 2 son españoles. Estoy seguro de que los avances en la medicina e investigación médica que nos van a presentar permitirán mejorar nuestra comprensión de las enfermedades. Nuestra fundación seguirá trabajando en esta dirección, apoyando proyectos de investigación o favoreciendo el intercambio de experiencias y conocimientos entre expertos. Es un camino que seguiremos recorriendo con el MIT, con el que ya tenemos no solo una larga y exitosa colaboración, sino una amistad de verdad. Muchas gracias y tiene la palabra Klaus Schleicher.
Sí. Muchísimas gracias, Ramón. Encima hay una historia también sobre el nombre mío que es muy alemán, ¿no? Porque estuvimos, Eduardo y yo, Eduardo Garrido, en Chile un día y entramos a una oficina y me preguntaron el nombre y decía, había una señora mayor y me decía nombre y dice Klaus. Me ponía Carlos, digo apellido Schleicher y ponía Sánchez. Ahora me están llamando Carlos Sánchez. Bueno, antes de nada, no sé dónde el mando, es por aquí. Déjenme, hablando un poco de MIT, el castellano encima no es mi idioma, primero es alemán y es italiano, pero el castellano lo he aprendido con amigos y con familia, ¿no? Y en MIT tenemos solamente 1.000 profesores, 3.700 personas que están haciendo investigaciones y 12.000 estudiantes, ¿no? Somos una universidad de investigaciones y tenemos casi 100, me parece que son 99, ya son 99 premios Nobel, premios, gente que tenía un premio de Nobel. Y tenemos, me parece que son 7 u 8 que todavía están trabajando en MIT, ¿no? Tenemos 6 escuelas, ¿no? Son 6, ¿no? La ingeniería, la ciencia, arquitectura, la de management y de social sciences and humanity, ¿no? Esas son las 6. Y la escuela de computer science de Schwarzman Computing, ¿cuál es? Schwarzman College of Computing, así es. Y nosotros estamos preocupándonos siempre de los problemas del mundo. Queremos salvar los problemas del mundo y mejorar los humanos que viven en este planeta, ¿no? Y gastamos en MIT solo casi 900 millones de dólares para la investigación todos los años. Y tenemos casi 66 laboratorios que están preocupando sobre temas que provoca el mundo para pensar las cosas. Y cada año lo sacamos casi 400, 500 patentes y 25 hasta 50, hasta 100 compañías que lanzamos del MIT. Y antes de la pandemia, el Reuters hacía una investigación mirando a las 100 universidades del mundo cuáles son las más innovativas. Y MIT llegamos al punto 169 de una escala de 200. Así dicen que MIT es la universidad más innovativa del mundo. Y esos son universidades con las que comparamos en el mundo y las mayorías están en los Estados Unidos, como Harvard. Nosotros referimos siempre a la universidad que está arriba del río, muy cercanas de MIT. Y el efecto que tiene es el impacto que tenemos en MIT, que lanzamos muchísimas compañías. El campeón de lanzar compañías es Bob Langer, que ha lanzado casi 59 compañías hasta ahora. Y en total, es un mogollón de compañías que han salido de MIT, es casi parte de MIT de lanzar compañías. Y todavía, en efecto, hoy en día están 33.000 compañías que están produciendo casi 2 trillones de ventas todos los años. Así, el impacto económicamente con 1.100 profesores es impresionante. Y Dan Anderson y Mark Barth también, que van a presentar hoy por la tarde, también tienen unas startups y startups de MIT que van a presentar hoy por la tarde para enseñar cuáles son las cosas que salen de MIT, las startups que tenemos, ¿no? Y en nuestra oficina, que la pusieron en marcha hace 75 años, nosotros encargamos para hacer las conexiones con la industria y MIT. Casi somos personas que lo metemos en cargo para traer valor a la industria, porque la colaboración con la industria para nosotros es muy importante, porque ahí están los problemas reales. Y MIT es un sitio que atrae el talento del mundo que quiere resolver estos problemas. Y casi cuando las cosas no se pueden vender, no tienes el DIR para investigarlo. Son las cosas más importantes. Y las 3 cosas que hacemos bien en MIT es la educación, la investigación y luego el entrepreneurship, para teniendo esta tecnología y estas ideas que salen de MIT para meterlo en startups, en compañías que van a cambiar el mundo. Y cuando lo estás poniendo con las necesidades de la industria, ¿no? Que están buscando talento, que tienen problemas de ingeniería, de research and development, y están también buscando startups para invertir en ellos y colaborar con ellos, ¿no? Y nuestro programa lo hace que arrancamos reuniones con los profesores, con las startups, hacemos conferencias como esto, donde presentamos qué hacemos en MIT, qué son las inversiones nuevas, y ponemos a una persona en carga con cada compañía que trabajamos que está en el programa que se llama Industrial Liaison Program, para traer y representar los intereses de estas compañías en MIT, para haciendo las conexiones con las startups, con los profesores, con los laboratorios y qué está pasando en el ecosistema de MIT. Y Catarina Madeira, que es una colega que está con nosotros, luego va hablando un poco más de las startups y va presentando las startups que hemos traído por aquí. Y Eduardo, que es mi colega, que es casi español, que viene de Argentina, la va haciendo la moderación. Así, muchísimas gracias y gracias a Raimundo, parte de todos aquí, y gracias por venir para estar con nosotros.
Buenos días a todos nuevamente. Bienvenidos al 7º Simposio Internacional de MIT Fundación Ramón Areces. Mi nombre es Eduardo Garrido. Yo soy Program Director de la Oficina de Relaciones Corporativas del MIT. Como saben, este simposio es sobre revolucionando la salud a través de investigación y tecnología de vanguardia. En los últimos años se han producido significativos avances en la investigación médica, surgiendo nuevos tratamientos y tecnologías que están revolucionando el cuidado de la salud. Como bien mencionó Raimundo, algunos de estos avances incluyen la medicina de precisión para un tratamiento más específico y personalizado, la inmunoterapia para combatir las células cancerígenas, la terapia génica, la inteligencia artificial para mejorar el diagnóstico, el tratamiento y los resultados de los pacientes, y la nanomedicina para desarrollar nuevos tratamientos y herramientas de diagnóstico. Hoy tenemos el privilegio de contar con 3 muy destacados investigadores altamente reconocidos y 5 startups muy innovadores en ciencias de la vida y cuidado de la salud del MIT. Voy a arrancar con el primer ponente, el profesor Mark Bate. Mark Bate es profesor del Departamento de Ingeniería Biológica del MIT, director de transformación de la educación en nueva ingeniería del MIT, miembro de la iniciativa de la Escuela de Medicina de Harvard para Medicina del ARN y miembro asociado del Broad Institute. El profesor Bate dirige un grupo de investigación interdisciplinario centrado en ingeniería de ácidos nucleicos, como nanomateriales altamente programables para su aplicación en vacunas, terapias avanzadas, diagnósticos, vigilancia de patógenos, biología estructural y computación. Es cofundador de 2 startups, Caché DNA y Cano Therapeutics. Él disertará sobre materiales de ácidos nucleicos de próxima generación para la salud, Al final de su presentación van a tener un espacio para hacer 2 preguntas y después va a haber un panel donde van a poder extenderse en las preguntas por parte de ustedes. Profesor Bate, please join us.
In the back. It's showing up there in the back. It's just not here. Okay, muchas gracias. Buenos días. My name is Mark Bata. I'm going to be talking about nucleic acid materials for healthcare in thinking about next generation modalities beyond where we are today for medicine. Is it coming back or? Switch. Okay. Sorry for the slow start. So these are my disclosures, just co-founder of Kana Therapeutics and Cache DNA, both of which are startups out of our lab, and I'll talk about today as well briefly. So we saw a nice picture of MIT. Just wanted to start with orientation. So Boston is across the Charles River where we're coming from, and Cambridge has this address for MIT, which is going to come up later in my talk. The zip code is 02139 and 77 Massachusetts Avenue. Okay. I wanted to share also that we're well known in Boston and New England for football, which is an American term for a sport that we play with our hands. And we're also well known for our delicacies and our cuisine, in particular Dunkin' Donuts. So we're here today because we face innumerable incurable diseases. The genomic revolution has now identified over 10,000 genetic and rare diseases, 600,000 US and almost 2 million European deaths from cancer annually. And we also suffer from a number of infectious diseases, including HIV/AIDS, which have no cure currently and no effective vaccines, with over 40 million deaths to date worldwide. Therapeutic nucleic acids, which our lab is working on, have major potential for medicine. These are classes of nucleic acids shown here, short interfering RNAs, antisense oligos, as well as messenger RNAs and CRISPR modalities. These allow one to knock down gene expression or express specific genes, or even edit and change genes within the human body to alter function and cure disease. These are incredibly powerful modalities because genomics and personalized medicine have revealed the gene targets for a range of diseases, including cancers. And importantly, 85% of the proteome, meaning all the proteins in the body, cannot be drugged using small molecules, which we're used to, small chemical molecules. So there's a tremendous need in this space, and in principle, these classes of medicines, therapeutic nucleic acids, could cure or treat nearly all these diseases in principle. The challenge, however, is to deliver these modalities within the human body to specific organs and tumor sites. And there are 4 major delivery barriers to bring these medicines to the sites of disease to be effective. The first one is following injection. They have to passively biodistribute from the injection site to the organ of interest, for example, the brain or the CNS or a tumor. Once in that organ or tumor, they have to target the specific cell of interest that's diseased. They have to get into the cell and traffic within the cell intracellularly. And then they need to affect their gene therapeutic action in the cytoplasm of the cell or in the nucleus. So these are all major challenges for bringing these therapeutic nucleic acids to market for patients in need across all these different types of diseases. Now, viruses, such as adeno-associated virus, people may have heard of AAV, have solved this delivery problem in a remarkable way through evolution. And they're incredibly effective at reaching specific organs in the body, including the brain, as well as delivering their gene therapeutic cargo or their gene cargo to these cells to express genes. They achieve this by assembling what's called a biomolecular or protein corona, a shell around the viral particle that forms when you inject it in the body, in the blood, or subcutaneously. And then as they traffic to organs in a specific way, they also present multivalent epitopes or peptides that allow them to target and enter cells very effectively. So these have been explored in the clinic and continue to be explored in the clinic for decades now, but they have encountered severe limitations in their deployment towards therapies. Several of these are listed here. The first is that the gene cargo of the AAV is limited to a vector that contains around 4.7 kilobases of DNA. So they're not generally applicable to these other modalities of therapeutics, including siRNAs, messenger RNAs. In addition, many patients have pre-existing immunity to this virus because we've seen it before. So we neutralize the virus upon injection with antibodies, and that requires immunosuppression for months for people to be able to tolerate these and also be able to deploy them in patients. Third, patients exhibit immunotoxicity that's unpredictable. There was recently a patient death that was published in the New England Journal of Medicine a few months ago, where a patient responded to a high dose of AAV with innate immune reaction, went into shock, and unfortunately died. And finally, the manufacturing cost has been quite high. One might argue well beyond the reach of most patients in the world, even in the West, spanning hundreds of thousands of dollars up to millions of dollars. Clearly not a therapy that's going to make it out globally for impact. The other modality that's been explored, and very successfully in a few cases, is lipid nanoparticles, pioneered by a number of folks, including at MIT, Bob Langer and Dan Anderson, who's here. The advantage of these is that they're low cost compared to viral vectors. They protect and encapsulate the therapeutic cargo of interest, the messenger RNA or the siRNA. And they also facilitate release of the cargo within the cell in the endosome when the pH drops. So very powerful, and have impacted the clinic as well with several licensed drugs as well as vaccines. They also have several disadvantages, however. So far, they've exhibited limited organ and cell targeting, so they mostly go to the liver. It's been challenging to replicate what viruses can do, for example, CNS targeting or brain targeting. They are limited in their dosage, also due to toxicity that can arise. And then finally, they have heterogeneous or variable composition. So viruses are molecularly identical. These particles can vary in their composition and be challenging to characterize as well, especially if one makes more complicated medicines, including multiple payloads. So in our lab, it's clear that we have this observation that both AAVs and LNPs have their own powerful capabilities and strengths. And it's clear that we would like to develop next-generation delivery tools that leverage these strengths of these modalities, but overcome their limitations. And so in our group, we're exploring nucleic acid nanotechnology for this purpose, in which we take messenger RNA or DNA and fold it into specific virus-like particles, shown here. These are either made of DNA or RNA or mixtures of the two. This is the same size as an adeno-associated virus, and it has the same structure. It's a 25 nanometer icosahedral particle, but it's not a virus. It's nucleic acids, and we can functionalize it and tailor its properties to be able to mimic aspects of the virus. So what I'm going to show you in the next few slides is foundational work towards building this platform to enable translational medicines, although it's still a few years off before we are really able to leverage this modality, but I'll show you the progress we've made to date. So the first question that we encounter, of course, is whether we can produce these particles at clinical scales, and the answer is yes. So the way we make these is with a long single-stranded DNA or messenger RNA that we fold with complementary oligos that are solid phase synthesized. Those oligos are produced in the same way that current drugs from Alnylam, for example, are produced, with solid phase synthesis. We functionalize those using covalent chemistries, and this long single-stranded scaffold strand, which we use to fold the particle that forms the template, we produce in an E. coli bacterial cell with helper plasmid, it's called, that allows us to ferment and grow very large quantities of this scaffold in a fermentation process that's fully scalable to clinical scale. So that leads us to the first startup opportunity that we saw in developing this technology. So because we were able to engineer custom single-stranded DNA scaffold of the custom length and sequence, we saw an opportunity to leverage this for cell and gene therapeutics ex vivo as well as in vivo, and formed Kano Therapeutics. And people are familiar that DNA is a template for cell and gene therapeutics, for example, in CAR T therapies, as well as other therapeutics. And the challenges in the field have been how to produce high quality and gene length DNA for these purposes. Chemical synthesis is one approach, but makes short oligos and are not easily purified. Enzymatic approaches have emerged recently using a variety of enzymes, but they also are challenging and costly to scale. Whereas with microbial fermentation, it's using a natural organism, in this case it's M13 and E. coli, which allows you to grow very large quantities and purify them in cGMP quality. And so results from Kano Therapeutics show, in comparing the circular single-stranded DNA, that's the template that we produce, to linear single-stranded DNA and double-stranded DNA in an application to a CAR T therapy, called BCMA here. The circular single-stranded DNA is much less toxic on the one hand compared to linear double-stranded DNA. So this is showing the cell counts. So low cell counts at high doses means the cells are dying. In the middle, what you can see, however, is that circular single-stranded DNA is also much more efficient at gene insertion than single-stranded DNA. And so on the far right, what you see overall is that this template that we make, the single-stranded DNA, is both low toxicity and considerably higher efficiency in gene insertion in this T-cell application. And so the other strength of this is that one can deploy sequence variability in the biological organism, which is not possible with enzymatic or solid phase synthesis. So one can evolutionarily select from libraries of this organism that creates diversity in the single-stranded DNA sequence in order to basically build in high-throughput screening to look at insertion efficiency, for example, in T cells, and then once one has the sequence of interest therapeutically to scale that up in production. And so this startup is founded by Floris, who was a former postdoc in my lab, and has been funded by the MIT Engine with Ann DeWitt on the board, and also recently recruited Brian Shai and Stephen Sofin, experts respectively in CRISPR gene engineering from UCSF and in scalable production of therapeutics, including at CRISPR Therapeutics and Avata as scientific advisory board members. Okay, I think we lost. Let me go back to this. Hopefully the debugging will help for the next talks. Right up there, I can see it. So. Okay, looks like we're back. So the next question for these particles, now that we can produce them at clinical scale, is whether they reproduce the properties of viruses in multivalent virus-like cell binding. And so, so for this application, I'll just note that there's a long history of vaccines that are clinically approved that consist of virus-like particles that are inactivated or recombinantly made. And this has been a very important way to stimulate humoral immunity. So many of these vaccines we get when we're babies, for example, for measles and mumps and rubella, and they last a lifetime following boosting. And that's because the multivalent nature of these particles stimulates B cells that then also create memory cells for our immune system to be able to produce antibodies for life. And so we tested this material for this property by focusing on the SARS-CoV-2 antigen called the receptor binding domain of SARS-CoV-2, where we display the protein fragment, that's the antigenic fragment of interest, onto the virus-like particle in order to test its ability to bind cells and activate B cells, both in vitro and in vivo. And this is a data-heavy slide, but the fundamental observation here is that both in vitro and culture, these particles bind the ACE2 receptor that's of interest for this viral-like protein. And if you look on the right-hand side, as you change the density of these antigens on the surface of the particle, what you see on the bottom is that B cells, which are the cells that first recognize these particles, are increasingly activated due to this higher copy number. So the answer is that yes, these particles reproduce the multivalent nature of a virus-like particle displayed here with the SARS-CoV-2 antigen. The next question that we faced is whether these particles, because they're made from nucleic acids, are going to be stable and functional in the body in vivo, because those were in vitro data that I showed you. And the answer is yes here as well. So this is showing a wild-type mouse administration where we inject and boost together with collaborators at the Raygon Institute. And what you can see on the right is that the IgG titers that are elicited in this mouse model are specific and strong to this receptor binding domain from SARS-CoV-2. And there's also this valency effect where the 30 copies give you a higher immune response than the six copies only. And this virus both protects in a challenge study that we've done with Michael Diamond at Washington University, but also generate titers that you can see here on the right that are significantly more neutralizing of the virus. So these are high-quality antibodies in addition to robust antibodies. Then the next question is whether these nucleic acid virus-like particles produce antibodies to the actual scaffold itself. So this is a major problem, as I mentioned, for AAV and other protein-based materials, because if our immune system recognizes those protein-based materials, it can neutralize them, and you can't then use that for a gene therapeutic delivery where you administer a medicine every month or every 6 months or every year, because your immune system neutralizes that, and that becomes ineffective. And so this observation then was to compare these particles to a protein-based particle, where what we found is that on the right-hand side, you can see for the protein particle, in this case it's a ferritin protein particle, you have antibodies to that protein scaffold in addition to the receptor binding domain of interest, this antigen, whereas in the case of the DNA virus-like particle, you do not get those neutralizing antibodies. So that's a very important observation for the deployment of this material in gene therapeutic delivery where one has multiple administrations, because it means that the immune system doesn't recognize the carrier as it does for protein-based particles. Finally, we also have looked at the immunotoxicity of these particles or materials at clinically relevant doses, and there's a lot of data here as well, but basically what you can see is that in looking at high dosage intravenous to the mice, the body weight stays the same. All the liver enzyme readouts of toxicity are normal, so there's no effect of this material that remains sort of in the blood and circulating without causing toxicity. And there's also, so far, not been seen any innate immune response, as you see with the AAV delivery materials. So far, and other labs have reproduced this as well, so far these materials look like they don't elicit antibodies that neutralize them, and they also are tolerable at clinically relevant doses. Finally, an attractive feature of these materials is that one can incorporate adjuvancy, so one can incorporate what are called CpG domains that act as adjuvants that help stimulate the immune system, which helps with vaccine applications, where you can increase the copy number shown as the red dots in the bottom of these CpGs and increase the immune stimulation in a quantitative way as a programmable medicine. So that's also a very unique feature of these particles that, again, resemble these virus-like particles. So in summary here, we've achieved scalable production of these materials with similar cost to therapeutic nucleic acids that are in the market. These have favorable sizes for biodistribution and multivalent display to engineer the protein corona. These can be loaded with arbitrary therapeutic nucleic acid cargoes, including those listed here, messenger RNA, ASO, and sRNA, as well as single-stranded DNA cargo for homology-directed repair and CRISPR gene template editing applications. They're also biodegradable, and they don't generate these anti-double-stranded DNA scaffold antibodies that would neutralize them, and they also have shown limited immunotoxicity. So for us, this sets the foundation for the future of delivery using these particles. And here's where the address of MIT comes back into the picture. So engineering the protein or biomolecular corona the way viruses do, which we're doing with these materials, allows us to get into Cambridge with the 02139 zip code. So that's a little bit like getting into the tumor of interest or into the organ of interest, for example, the brain. And then these multivalent ligands that we display, where we have this virus-like properties of these particles that are very attractive for cell recognition, allow us to get to the actual address of MIT and home into the specific cell, which is like walking to 77 Massachusetts Avenue, which is the actual targeting. So both of these components are needed, we believe, for mimicking the behavior of viruses in their ability to target arbitrary cells and tissues within the body for gene therapeutic action. So to summarize, the therapeutic nucleic acids really we think of as 0.0 was their discovery, which has now resulted in 3 Nobel Prizes across the discovery of RNA interference, CRISPR, and mRNA. The version 1.0 is the current medicines and vaccines that are in the clinic. These largely rely either on GalNAc, a small sugar molecule that allows these medicines to target cells in the liver, or lipid nanoparticles that also go to the liver or are used for messenger RNA vaccines, for example, in SARS-CoV-2 or COVID-19. But the future, a decade away from now, is going to be using these types of medicines to deliver outside of the liver to tumors and organs, with a very important organ of the CNS where Alzheimer's can be treated, as well as Parkinson's and a number of other neurological diseases. And there's, of course, a tremendous need in other organs as well, but the CNS is what we're most focused on in our lab and deploying this material going forward. So shifting gears from therapeutics to diagnostics, nucleic acids as next-generation materials are also playing a very important role in this space, as people are familiar with, in exosome diagnostics, and of course we're all used to diagnostics with COVID-19, for example, to get a nasal swab where then the nucleic acid is diagnosed to be either positive or negative for SARS-CoV-2. So once again, I apologize for some reason having exited. So a high-level analogy in this space, both for therapeutics and diagnostics, is really the shift that we're going to be seeing in this century from electronics to store information towards nucleic acids, both DNA and RNA, for storing information. So of course, nature has been storing information in nucleic acids for billions of years. That's our genetic information, our genomes, that we're very familiar with. But there's a parallel field that's been using DNA, which is what our lab started with, to store digital information. For example, an ATGC sequence can be converted into zeros and ones. The same way as you have a file in your computer, that could be an image or a movie, you can write that file into DNA. And originally, we were motivated to try to help solve this problem because it turns out that the data centers that we're using currently to store all of our movies and images from Facebook and Google and so forth, and our iPhones, cost billions of dollars to build and maintain. They're incredibly energy hungry. They use hundreds of megawatts of energy. and they also require several acres of land. So this Google data center stores around an exabyte of data. An exabyte of data is 1 million terabytes. So that's about 1 million laptops. So it's a lot, but on the planet today we have about 100 zettabytes, where a zettabyte is 1,000 exabytes that we generate every year of new data. So the amount of data on the planet is exploding, and the question is, can we use other materials to store information? And an interesting fact is that DNA, because it's so dense, the size of the DNA Watson-Crick base pair, it could store an exabyte of data if a flash drive were made from DNA. So that's a remarkable fact. That entire Google data center shrunk down into the size of a USB hard drive if it were made from DNA. That's the density of genomic information in our cells. The other attractive feature of DNA is that if you think about our conventional data storage media, whether it's tape drives or solid-state drives, typically after 5 or 10 years, they fail or have to be replaced, whereas we have evidence of DNA lasting over 1 million years from a Siberian woolly mammoth in the permafrost. So DNA is not only incredibly dense as a storage medium, it also can last effectively forever, and it doesn't require any energy once it's written, because once you make the DNA, you don't have to maintain it with energy and cooling and so forth the way you do with these other media and that Google data center that I showed you. The final point is just that DNA can be used to store information in materials such as pills and medicines where no other media can be used. So if you wanted to store, for example, patient data, regulatory information, or manufacturing data on a pill, you could do that by encoding that information into DNA and putting the DNA within the pill, which you can then read out with sequencing. So that's something you would never be able to think about doing with conventional data storage media shown here. So there are a lot of startups and technologists that are working on developing this further, and there's very large players in the United States that are working on this, including Microsoft, who has a very active arm in DNA data storage, as well as Western Digital, and Dell is also exploring this. But I'm not going to be talking about that other than the technology that we developed to try to help solve this problem, which spun into then a genomic storage platform. So what we developed is little mini capsules that are the size of exosomes or the nucleus of a cell, roughly a micron, which is 10 to the minus 6, so 1 millionth of a meter, where we encapsulate genomic data, in this case RNA as well as DNA of organisms. For example, a SARS-CoV-2 swab from your nose can be encapsulated in this particle, and then we label it with nucleic acid barcodes that tell us the information of what is inside of that particle. And so, for example, if you thought of this as the way Google stores images, they have information about where the image was taken, the date, the time, and so forth, and what's the content of the image. For us, we also could store the image, like of a cat, using these types of labels, orange, cat, and domestic. And the important point is that with 4 of these barcodes, we can label 10 to the 20, which is many, many, many orders of magnitude beyond a billion, unique such particles in a very small footprint that would fit in the palm of my hand because these are so small. And so if you think about that in the genomic space, where you're storing SARS-CoV-2 samples or human patient samples, you have an incredible density and capability to store genomic information that we currently cannot store. And so this is a little bit like thinking about, today we use freezers and fridges to store genomic information, if we store it at all. And what we're doing is turning that genomic information into what would be a Google of books, where we can use this encapsulation strategy with barcodes to search and retrieve arbitrary samples of nucleic acids, whether it's from SARS-CoV-2 testing or from cancer or reproductive health testing in genomics. And so this led to a second startup, which will be speaking later this afternoon, and these slides are courtesy of Heather, who will be presenting later today, where we spun out a company called Cache DNA. And James was a former postdoc in my lab at MIT, and here you can see the mission of the company. And so as I said, today we're familiar with whether it's blood draws or nasal swabs, there's genomic information being collected and tested for reproductive health, for cancer biopsies, and for rare disease testing, using a variety of approaches that we're familiar with. And the challenge really is where and how do you store all of this information? It's much too expensive to sequence it all, so typically it's thrown away if it's stored at all. For a short term, it will be in a freezer, for example. And this shows an example of this. So these freezers can occupy warehouses where you might store millions of samples. But of course, we have billions of people on the planet, and the goal of genomics is really to collect all that information from humans to be able to understand rare diseases and variants. And this solution will not take us where we want to go. And so a warehouse of freezers like that, as I mentioned, which is millions of genomic samples, could fit in the palm of my hand effectively with the solution of this microcapsule tagging that Cache DNA is developing. And you can think of this as nature currently uses exosomes, or you can think of a nucleus to be able to encapsulate these materials. We use, in our lab, we used a glass, and in Cache they're developing polymers with these DNA barcodes that are part of the invention to be able to access this very dense and complex information, where here it's now genomic information from samples that are collected. And that's all stored at room temperature, doesn't require freezers in addition, or any energy, in addition to the fact that it has an incredibly dense footprint. And so these are some quotes that Heather collected from different companies and institutes on what the actual bottleneck is. So we're humans, we like to save everything. If you look in your basement or your closet, you typically don't throw things away. And in our iPhones, we like to save every picture we have in every movie. The same is true for genomic information. We'd like to test everywhere and all the time to track pathogens, for example. These could be SARS-CoV-2 or new emerging pathogens that we haven't yet detected. In airports, in schools, in nursing homes, we'd like to collect all of that information so that if there's another pandemic, or when there's another pandemic, we have the ability to go back and search through that information to be able to sequence and understand where that pathogen came from and how it evolved. Currently, the state of the art is for the CDC in the US at least, if there's an outbreak, to go out into the field, into airports and so forth, and do active testing to try to understand what happened. Storing all of that information would allow you to analyze all of that. But currently, it's much too expensive to do that, and like I said, it takes up much too much space. And so there's a huge bottleneck in this area. And most companies, as well as institutes, are solving this problem in-house with freezers and other solutions that they can find. And so Heather will talk more about how Cache DNA is partnering with a variety of institutes and companies to be able to deploy this technology and pilot this technology for the diagnostics space using this innovative nucleic acid barcoding approach. So that wraps up my presentation. So what I wanted to share today is a couple of different next-generation nucleic acid nanotechnologies, one in the therapeutic space and the other one in the diagnostic space, that over the coming decade or two may revolutionize our abilities, both to cure diseases with messenger RNA and siRNA, by leveraging their ability to look like viruses, to mimic viruses for in vivo targeted delivery, although it's early days, and we will see how this plays out. And then secondly, to be able to really detect, diagnose, and prognose diseases by eliminating the bottleneck that we currently have around collecting massive, massive amounts of genomic information from humans as well as from pathogens, and also in preservation of planet Earth to collect all the species that we have on the planet, which is many orders of magnitude more than the data we have, to be able to catalog as we are undergoing a mass extinction. So I'd like to leave with the following quote, which some of you may have heard about from Roy Amara. He was the past president of the Institute of the Future. And what he said is, we tend to overestimate the effect of a technology in the short run and underestimate its impact or effect in the long run. So we're living through this with artificial intelligence right now, as well as other technologies, some of which I talked about. And if you look at any technology arc, including the internet perhaps most prominently, if you think about the late 90s, for those of you who are old enough like me, and the early 2000s, there was an incredible expectation of the internet, which then crashed quite significantly in the mid-2000s to late 2000s. But then the ultimate rise of impact of the internet now, today, in the 20s and 30s, is far beyond what we ever expected. And so if you think about other technologies, you'll typically see that arc as well, and I think it also applies to nucleic acids and next-generation medicines and diagnostics in this space. With that, I'll just thank, of course, the students in the group, as well as really important collaborators that we've been working with at MIT, as well as at Stanford, the funding, and of course, the Fundación for the opportunity to come and speak here today. I'd be happy to take any questions that there are. Okay.
I think va a ser mejor que las preguntas las hagamos en el panel que viene después, al cierre de las presentaciones. Así es. Thank you.
Yeah, thanks.
Me gustaría ahora presentar a la siguiente speaker, la doctora Mercedes Balcells-Camps. La doctora Balcells-Camps es investigadora científica principal en el Instituto de Ingeniería y Ciencias Médicas del MIT y profesora titular de bioingeniería en el Institut Químic de Sarriá. ¿Lo dije bien? Su investigación en ingeniería de tejidos ha demostrado cómo los estados de las células endoteliales son fundamentales para la respuesta del tejido a una lesión. Ella fue la primera en demostrar que las células responden a la frecuencia del flujo, no solo al esfuerzo cortante, y de una manera que depende del hecho del tejido. En 2011, el Gobierno de España le otorgó la Cruz de la Orden del Mérito Civil por su contribución a establecer un canal fructífero entre el MIT y España. Además, preside el programa de innovación del MIT Idea to Global dentro de la iniciativa LinkU del MIT. Ella disertará sobre comprendiendo la interfaz entre la salud vascular y mental desde la nano y la microescala hasta los tejidos, órganos y pacientes. Doctora Balcells.
Bueno, muchas gracias, buenos días. Gracias, Eduardo, por la introducción y a la Fundación Ramón Areces por tenerme aquí. ¿Sí? Bueno, perfecto. Vale. Gracias por la introducción tan amable, Eduardo. Yo prefiero presentarme de una forma más personal. Nací en Barcelona, soy catalana. ¿Alguien me ha dicho que tendría que hablar en catalán? No, prefiero hablar en español. Crecí viendo estos andamiajes en la Sagrada Familia y nunca pensé que un andamiaje sería algo con lo que trabajaría cada día. Y bueno, decidí estudiar química gracias a mi profesora de, en aquella época, séptimo de GB, que estábamos todos aprendiendo la tabla periódica de los elementos y estábamos todos así dormidos, aburridos, ¿no? Memorizando, y de repente dijo algo que me cambió la vida y dijo: la tabla periódica de los elementos es solo la punta del iceberg, lo que está debajo es todo lo que queda por descubrir. Y aquel día yo dije: voy a ser química y voy a ser investigadora. Así que si hay algún maestro en la audiencia o algún padre como yo de niños más jóvenes, cuidar mucho lo que decís, porque una frase te puede cambiar la vida. Y sí, soy la mayor de 6 hermanos. De hecho, estoy aquí con mi mayor fan y animadora, mamá. Mi mamá ha venido desde Barcelona para verme. Mamá, levanta. Y como la gente del escenario y del mundo del espectáculo dedica las cosas a sus fans y a quien quieren, pues yo hoy le voy a dedicar esta charla a mi madre. Hice mis primeros experimentos muy poco ortodoxos, mis primeros clinical trials, ¿no? Pasaba muchos domingos en casa de mis abuelos, que tenían un jardín, y yo cogía todas las plantas, frutos, imaginaros todo, hacía pócimas y se las daba a mis hermanos pequeños. Entonces, claro, mi madre se quedaba sorprendida y decía: ¿cómo es posible que todo el mundo tenga diarrea menos Merche? Bueno, ya lo sabéis, yo no tomaba mis pócimas y la buena noticia es que todas las plantas y verduras y todo del jardín de mi abuela no hay nada letal. Cuando hablo y digo que hago ingeniería de tejidos, mucha gente se piensa que trabajo para Inditex y de hecho tengo una hermana, María, que se ocupa de que siempre me viste guapa para presentar y ella trabaja para Inditex, pero no son estos tejidos en los que yo me dedico. Y cuando hablo en inglés y digo que hago tissue engineering, la gente se piensa que hago tissue paper, ¿no? Papel de váter o de Kleenex. Tampoco. No vamos por ahí. Mi campo de investigación se basa en usar células humanas con unos andamiajes, ¿no? Como os decía, que esas células que forman tejidos sólidos, la mayoría, son capaces de engancharse, adherirse, proliferar y formar un órgano o un tejido de reemplazo. La tercera composición que necesitamos es el entorno mecánico, ¿no? Porque nos podéis imaginar, por ejemplo, bueno, yo siempre estoy moviéndome, ¿no? Una rodilla, unas células de rodilla están sufriendo compresión, ¿no? Pues no te las imaginas quietas y paradas. Y lo mismo pasaría por la sangre que circula por vuestras venas y arterias. No te imaginas una arteria quieta. Entonces, con eso, pues hacemos... Mi aspiración, mi ambición es tener órganos y tejidos de reemplazo para aliviar las listas de trasplantes, que están las listas de espera de gente esperando un trasplante. La realidad es que son unos buenos modelos, ¿no? No son órganos perfectos, pero se acercan mucho para poder entender la enfermedad, para poder entender incluso cómo dos enfermedades interaccionan una con la otra. Así que hoy os voy a explicar un poquito cómo se puede entender la enfermedad cardiovascular y la mental a través de estos dispositivos o de estos constructos. Casi intuitivamente podéis pensar, ¿no? Algo que la enfermedad mental y la enfermedad crónica están interrelacionadas. Pensar en esto, ¿no? Tenéis una enfermedad crónica, como por ejemplo puede ser enfermedad de riñón, ¿no? Yo tengo un amigo muy bueno en Boston Medical Center, el doctor Chitalia, que dice que se encarga de esto, de estar en la unidad de trasplantes de riñón. Y la gente está en diálisis. Pues estar en diálisis, no sé si hay alguien en esta audiencia que tiene a un familiar con diálisis, es una muerte en vida. Tener que irte a hacer diálisis cada 2 días. Y al final me dice que sus pacientes no se mueren esperando un riñón, se suicidan, ¿no? O sea, mirar, ¿no? Y al revés, si uno le diagnostican con una enfermedad mental, como por ejemplo una depresión, pues empiezas a hacer cosas que no deberías, ¿no? No haces ejercicio, comes mal, bebes alcohol y acabas muriéndote de un infarto, no de la depresión, por ejemplo. Así que hay una relación, ¿no? Que uno lleva al otro y el otro lleva al uno. Y de hecho, si mis alumnos no se fiaban de mi intuición y hicieron una búsqueda bibliográfica analizando el lenguaje natural de todas las bases de datos existentes en medicina y encontraron hasta más de 7.000 estudios publicados que realmente corroboran eso, esa comorbididad, ¿no? Que hablamos, es decir, 2 enfermedades que se presentan a la vez y que no sabes muy bien si una causa la otra o la otra la una. Y esta es la pregunta que a mí no me deja dormir desde hace 15 años. ¿Hay alguna manera mejor de monitorizar y llevar, ¿no? A los enfermos que tienen enfermedad crónica y enfermedad mental o? Esa es la pregunta. Y si miramos en esa búsqueda bibliográfica que salían más de 7.000 estudios, pusimos comorbidad, monitoring, o sea, unos cuantos keywords y nos salieron solamente 5 estudios clínicos primarios. Así que poca evidencia de que esto sea posible a día de hoy. Esto no nos desanimó, al contrario, nos animó aún más, porque empezamos a pensar si sería posible aprovechar los modelos de vasos que os comentaba, ¿no? De tejidos, arterias, venas, capilares, para identificar y cuantificar moléculas que están en la circulación para identificar enfermedad. Y, además, aprovechar datos que generamos continuamente y que llevamos, como por ejemplo con relojes inteligentes. Perdonad, a veces estoy traduciendo en mi cabeza, ¿eh? Es que no doy esta charla muchas veces en español. Bueno, pues aprovechar la tecnología móvil que tenemos todos para entender el comportamiento humano. Y, bueno, empezamos a trabajar con un equipo a intentar fabricar arterias artificiales, aunque muchos médicos todavía no me dejan llamarlas arterias artificiales. En inglés es muy largo, me dejan decirle vessel-like structures, que en español con más palabras, algo que se parece a un vaso. Nos basamos en imágenes de pacientes, hacemos una interfaz computacional que nos permite reproducir, a ver si soy capaz, aquí, sí, cómo va el flujo por esa arteria. Imaginaros, ¿no?, que vosotros, a mí me gustan mucho los deportes de riesgo, pues estás haciendo rafting, ¿no?, y llegas a un momento, ¿no?, que hay como un remolino y te quedas allí parado. Pues así es lo que pasa en las arterias cuando hay bifurcaciones, hay zonas donde el flujo se queda más estagnado, ¿no?, y esas son las zonas problemáticas, donde empieza la enfermedad cardiovascular. Además de tener esas predicciones matemáticas que son muy útiles, imprimimos en 3D o por otros métodos todo tipo de andamiajes. Aquí solo es el material polimérico, que puede ser biodegradable, que puede no serlo, y con diferentes geometrías que realmente recapitulan las arterias. En este caso, pues mirar la de abajo, esta es una arteria carótida, esta es una arteria coronaria, e incluso esta coronaria con la forma, ¿no?, que las coronarias son las arterias que abrazan el corazón, así con ese ángulo. Esto solo es, como decía, el andamiaje, ¿no?, lo que da el soporte a las células. Son materiales de muchos tipos, no voy a entrar en detalle, pero que podemos regular la porosidad, ¿no?, para favorecer que las células crezcan dentro de ese scaffold que decía, y vamos creciendo el vaso capa a capa, ¿no?, como cuando haces un pastel o una lasaña, una capa, y por ejemplo en el caso de las arterias coronarias pues necesitamos células de fibroblastos, necesitamos células musculares, necesitamos células endoteliales, todas son no lo he dicho, todas son células humanas. Esto es importante porque muchos de estos tejidos, otro de las aspiraciones secundaria, pero que para mí es muy importante, es aliviar la experimentación en animales, al menos reducirla significativamente, porque si tienes algo como células humanas que recapitula la fisiología humana, a lo mejor podemos dejar de usar cerdos, que en el caso del sistema cardiovascular es el modelo más similar al hombre. Así que hablaba de los tubos, tenemos las células y el tercer ingrediente que os comentaba es el estímulo mecánico. Entonces, mi laboratorio es como un gimnasio, a ver, ¿cómo os lo explico? A veces es como un gimnasio para células y a veces es como una sala de torturas. Llego al laboratorio y mis alumnos me dicen, doctora Balcells, no ha quedado ni el apuntador, sala de torturas. Bueno, son dispositivos que llamamos bioreactores, que es realmente para reproducir el entorno mecánico o biomecánico a la que estos, en este caso, arterias deberían ver, ¿no?, cuando estuvieran implantadas. Así que veis, son grandotes, son del tamaño de una nevera, ¿no?, pero bueno, permiten conectar, aquí lo veis, ¿no?, pues con varias réplicas, 14, 24, ¿no?, pues conectar estas arterias artificiales y que crezca el tejido de forma como si estuvieran, pues, como si fuera una coronaria en el corazón. Le podamos dar pulsatilidad, le podamos dar la fuerza de rozamiento simulando el fluido de la sangre. Y también lo bonito es que yo hago arterias sanas, pero también podemos hacer arterias enfermas, porque podemos, por ejemplo, simular una arritmia, o poner azúcar en la sangre, o poner una inflamación, y así, ¿no? Por eso decía que es cámara de tortura a veces, porque si te pasas demasiado, pues no queda ninguna célula, hemos pasado la tolerancia del sistema. Aquí tenéis pues, otra demostración, ¿no? Las células, para poderlas ver, las tenemos que teñir con diferentes marcadores fluorescentes, pues mira, los fibroblastos, las aórticas, musculares, las endoteliales. Aquí tenéis el scaffold ya montado. Y los médicos que os decía nos dijeron, bueno, Mercedes, que tengas células capa a capa en el lugar que toca está muy bien, pero tienen que funcionar como una arteria, así que nos lo vas a tener que demostrar. Y nos hicieron pues, un año de experimentación demostrando que podían contraerse, dilatarse, que si les hacías una inflamación se inflamaban, que si les hacías una herida coagulaban, etcétera, etcétera. O sea, esto es solo una tabla que resume, no voy a entrar en detalle, que realmente conseguimos convencer y publicar que estas arterias realmente se parecen mucho, mucho, mucho. Bueno, he hablado de enfermedad cardiovascular, arterias, o sea, tenemos una herramienta que se parece mucho a una arteria y podemos entender su patología. Voy a ir ahora más pequeño, hacia el cerebro, porque os he dicho que hablaría de enfermedad mental y claro, para ir hacia el cerebro hay que hablar de capilares, o sea que vamos a ir desde el entorno macro, porque una coronaria tiene 3 milímetros de diámetro y lo puedes coger con tus manos, a el cerebro. Ahí el desafío fue muy grande, porque hacer un capilar del cerebro, y no solo un capilar, sino la barrera hematoencefálica, ¿no? Ramón y Cajal fue nuestro único Nobel español que definió esa barrera hace más de 100 años. Inyectó un colorante en la cola de un animalito y vio que todos los órganos se teñían de ese color, no sé si era azul, menos el cerebro que quedaba, ¿no? Y definió que había una barrera que nos protegía y por eso no nos hemos extinguido. Y por eso cuando tienes un cáncer en el cerebro o una infección es tan difícil llegar, ¿no? Es una barrera que nos protege, pero por otra parte es muy difícil penetrar. Entonces, crear un modelo de barrera hematoencefálica, en este caso necesitábamos células cerebrales, astrocitos, células endoteliales de los capilares y, de nuevo, tener ese flujo, ¿no? ese estímulo mecánico de la cizalla, para que la barrera fuera realmente barrera. Así que nos pusimos a trabajar con estudiantes, pues, Gonzalo Muñoz, que hizo su tesis conmigo, y diferentes tamaños, diferentes materiales, en este caso bioinspirados, colágeno, ácido hialurónico, mezclados con diferentes concentraciones, y con ese tamaño de poro diferente, pero también diferente crosslink, o sea, diferente reactividad entre las moléculas, ¿no? Para poder jugar y buscar unas condiciones de stiffness, o sea, una rigidez apropiada, ¿no? Aquí ves, por ejemplo, ¿no? Alguno que más que un hidrogel parece, pues eso, se nos desmontaba, se nos desmoronaba la estructura, eso no nos interesaba porque acordaros que tenía que poner luego flujo por encima. Pero bueno, llegar al rojo, aquí la barra roja, algo que tuviera la misma consistencia que el cerebro y en el que pudiéramos sembrar astrocitos humanos y que esos astrocitos, que a mí cuando los veo en el microscopio todavía no tengo astrocitos impresos en mi habitación, full closure, pero sí que en el laboratorio algunas fotos de estas sí que las imprimimos. Bueno, los astrocitos tienen esa forma de estrella, pero claro, depende de dónde, en qué material los imbedas, cambia su morfología y eso te dice mucho, ¿no? La apariencia, ¿no? Dice mucho. Así que tuvimos que ver cuál de estas estructuras no solo era se aguantaba el flujo, sino que teníamos astrocitos con la forma correcta, ¿no? Con esa estrella. Además, necesitábamos recubrirlo con células endoteliales, que es, si os imagináis el capilar, sería la parte de dentro, las células que están en contacto con la sangre. Así que conseguimos, aquí tenéis una imagen de confocal, donde tenemos en verde las células endoteliales de cerebro y en rojo los astrocitos. Y como necesitan ver flujo, pues creamos una pequeña cámara de microfluídica donde se puede colocar el hidrogel con las células y conectarlo a un bioreactor para que esas células endoteliales estén con las condiciones que verían si estuvieran en un capilar del cerebro. Y con esto podemos de nuevo estudiar, lo más bonito es que puedes estudiar el cerebro sano y luego crearle un insulto mecánico, por ejemplo, en este caso darle una sobrepresión. Imaginaros a alguien con hipertensión, ¿no? Y entonces aquí, ¿no? Os voy a enseñar las fotos bonitas, las sanas serían estas dos del medio, la B y la C, donde veis una proteína en verde, lo que sería el perímetro de estas células, es una proteína le llamamos zona occludens 1, ZO1, que es una de las proteínas encargadas de hacer ese efecto de barrera entre las células endoteliales y dejar que nada pase, ¿no? Pues, por ejemplo, si queremos una hipertensión y una cizalla mayor, y aquí veis en la foto en el C, pues esa barrera se ha roto, ya no veis el perímetro de estas células. Y si simulamos un stroke, un infarto cerebral, y paramos el flujo, que sería el caso del A, veis la barrera todavía, pero muy cerrada, hace zigzag. O sea, si haces zoom, ves que allá también hay hueco. O sea que podemos, de alguna manera, simular barrera hematoencefálica sana y enferma. Y ahora me preguntaréis, ¿y esto para qué sirve? ¿No? Porque claro, si yo ahora voy y le pregunto, a ver, déjame, alguien que conociera ayer, a Carlos lo conozco, déjame, ¿quién? Mira, te acabo de conocer, ¿cómo te llamas? Teresa, soy Mercedes, ¿te puedo abrir la cabeza y mirarte tu barrera hematoencefálica? ¿Ves? Gracias. Alguien que sí que me conoce, Carlos, déjame abrirte la cabeza y mirar tu barrera hematoencefálica. No, gracias, ¿no? Vale, pues estoy de acuerdo, estamos claros, ¿no? O sea, esto sirve para entender, es un modelo, para saber lo que estamos buscando, pero de afecto terapéutico no podemos ir abriendo cabezas para ver si vas a tener enfermedad cardiovascular y mental. No nos sirve, ¿no? Entonces, por eso hemos estado en los últimos años analizando qué producen esas células, ¿no? Y las células son fuentes de información increíble. Y lo que hemos encontrado es que segregan muchas moléculas, tanto si están sanas como si están enfermas, y que son dependientes de su estado. Y hemos estado, antes esto lo han hecho toda la vida, lo que pasa que es lo que decía, antes con un microscopio veías hasta aquí y ahora ves hasta allá, ¿no? Y estas moléculas son exosomas, que son moléculas nanométricas, o sea, imaginaros, cogeros un pelo, pues un millón de veces más pequeño que el diámetro de un cabello, ¿vale? No las veíamos porque también son tan pequeñas que tú centrifugas y si es una centrífuga normal no la puedes depositar y aislar. Pero ahora, usando una combinación de ajustar la densidad y una ultracentrífuga que es capaz de girar 150.000 revoluciones por minuto, pues podemos aislar estas vesículas, o estas exosomas que son nanométricos y que además los podemos separar por tamaño. Para que os hagáis una idea, estos agujeros negros de aquí, los señalo que es más fácil, ¿vale? Es el tamaño del papel de filtro, que tiene 0.22 micras. O sea, si estos son 0.2 micras, imaginaros el tamaño de esas bolitas blancas que son los exosomas y que varían en tamaño, y no solo en tamaño, sino que además dentro han encapsulado marcadores diferentes que estamos analizando, porque esperamos que nos den pista de lo que está pasando, ¿no?, en el cerebro, porque son capaces de pasar a la circulación sanguínea. Así que, y esto es work in progress, o sea, es lo que estamos haciendo ahora, tenemos modelos de barrera hematoencefálica, sabemos, ¿no?, crear una barrera sana y una enferma, estamos analizando todas esas moléculas, o sea, tanto moléculas como citoquinas de inflamación como exosomas, para entender cuáles de ellas son capaces de cruzar la barrera hematoencefálica y se presenten en sangre, y aún más ambicioso, se puedan coleccionar en algo que no tengas que pinchar a nadie, porque a mí es que me dan mucho miedo las agujas. Entonces, si pudiéramos detectarlas en saliva, en orina o en sudor, las podíamos cuantificar por métodos estándar de laboratorio, como un ELISA, pero la ambición, y tengo unos cuantos estudiantes de ingeniería eléctrica ayudándome a montar un sensor que lo podríamos detectar directamente, por ejemplo, imaginaros detrás de vuestro reloj, tener un sensor que pueda decirte o medirte estas partículas. Bueno, hemos pasado del mundo macroscópico al microscópico, al nanoscópico, ¿no? Y ahora os voy a llevar de vuelta al mundo de los humanos, ¿no? Las células segregan de forma pasiva exosomas que cruzan la célula de forma pasiva, y los humanos generamos de forma pasiva, sin darnos cuenta, millones de datos, porque llevamos un teléfono inteligente en la mano o un reloj inteligente. Así que podemos aprovechar esa parte también para compaginar o corroborar lo que nos dice la biología. Y bueno, para eso, parón. Yo sola no puedo hacerlo y MIT solo no lo puede hacer. Nadie lo puede hacer solo. Así que ahí me vi lanzada, obligada y con un gran placer a establecer una colaboración a través de varios programas internacionales que tiene MIT. Uno de ellos es MISTI, que es algo así como MIT International Science and Technology Initiatives. El programa MIT España, por ejemplo, que fundé y codirijo, es uno de ellos, pero tiene 36 programas internacionales MIT, y gracias al programa con México y al programa con España, pues pude empezar una colaboración. Por ejemplo, pues claro, si quieres conectar a inteligencia artificial, machine learning, cardiología, clinical psychology y sensores, ¿no? No lo tenemos todo nosotros y está bien así, pues aprovechar de estos programas. Y además del programa Idea 2 que mencionaba Eduardo, que también trae emprendedores a MIT de todo el mundo para que estas ideas y este conocimiento generado en el laboratorio pueda llegar a la sociedad. ¿Qué hicimos? ¿Qué empezamos a hacer? O una cosa que quizá no es obvia, ¿no? Pero que un teléfono móvil, todo el mundo tiene casi todo el mundo tiene teléfonos móviles en este momento, ¿no? En América hay más de 86 millones, en el 2019 ya había 86 millones y ahora ese número sigue subiendo. Usamos la información de ese teléfono móvil muchas veces, ¿no?, solo para nosotros, personal, pero también muchas veces, y las generaciones más jóvenes más que las más antiguas, para uso social. Pero entonces, ¿por qué los médicos? ¿Por qué solo hay 5 estudios clínicos que estudian el uso de sensores para que lo usen en la praxis médica. ¿Por qué cuando hablo con mis compañeros médicos no se acaban de creer? Mi jefe es cardiólogo y cuando le digo que con el móvil, con el teléfono, puedo ver mi ritmo cardíaco, me dice que esto no sirve para nada. Yo digo, ay, Elisor, ¿pero cuándo te lo vas a creer? ¿Cuándo te vas a creer que esto es posible, no? Bueno, pues el doctor Toros, hace muy poco, en 2016, definió algo que se llama digital phenotyping, que es, yo lo entendí así, esta es su definición, moment by moment quantification of the individual level human phenotype in situ using data from personal digital devices. Yo dije, a ver, que me lo expliquen. Mirad, el genoma, ¿no? Nuestro DNA es como cuando tienes el plano, sobre el plano, una cocina, ¿de acuerdo? Si al final viene el constructor y el dueño de la casa dice, pues yo quiero este tipo de suelo y estos grifos, este es el fenotipo. El fenotipado digital sería el equivalente en una persona a tener una cocina donde con un teléfono móvil tú puedes saber en cualquier momento, esto es fantástico, cómo funcionan si están funcionando todos tus electrodomésticos y si la nevera, esto es más importante para mí como madre de adolescentes, si la nevera está vacía. Pues eso sería digital phenotyping, es poder usar los datos que generamos para tener en cada momento, ¿no?, información clínica valiosa sobre tu salud. Y bueno, ¿cómo se hace esto? Pues bueno, en los móviles que comentaba hay muchos sensores, cada día van añadiendo más, ¿no? Sabemos que tenemos un GPS, tenemos un giroscopio, hay en la pantalla el botón de on and off, hay un micrófono, hay una cámara, etcétera, etcétera. Y todo eso son sensores que nosotros sin enterarnos están allá. Lo que se puede hacer es agregar varios de los datos de ese sensor y, por ejemplo, si combinas los datos de GPS con los del giroscopio, si te estás moviendo o no, puedes agregar y decir, esta persona está corriendo, está andando, está escalando, ¿no? Porque esos sensores te permiten decir dónde estás. O, por ejemplo, si combinas, aquí lo veo, el movimiento, no te estás moviendo, la pantalla está en off, ¿no?, y no hay mucha luz, pues puedes deducir que esa persona es su hora de ir a dormir y está durmiendo. Todo eso son características de un nivel, digamos, basal, básico. Más complicados, con esas características agrupadas, se puede llegar a tener patrones de comportamiento, de si estás cansado, si te concentras más, si has dormido mejor o peor. Todo esto lo sabemos y es lo que puedes a día de hoy, ¿no?, lo miras en tu teléfono o en tu reloj y sabes todo esto. Pero ¿veis todas estas líneas que he conectado? Cuando se trata de valor clínico, o sea, decir realmente estoy enfermo, tengo depresión, tengo enfermedad cardiovascular, no hay conexión. Falta llegar a ese nivel, ¿no?, para poder, por ejemplo, pues eso, como decía, monitorizar y controlar, no cuando vas solo al médico, sino de forma regular, esa enfermedad mental. Así que, con esa colaboración que os comentaba, y aprovechando algo que la verdad nunca me había pasado, que a una investigadora le cierren el laboratorio, porque yo hacía ingeniería de tejidos y no hacía nada con COVID, que te cierren el laboratorio un año por culpa de la pandemia es una desgracia, ¿no? Pero bueno, de esa desgracia pues me tocó cambiar el paradigma y empezar a pensar qué podemos hacer. Y el que podemos hacer es ensamblar un estudio que le llamamos de uno study, porque es el primero, habrá segundo y tercero, donde empezamos a reclutar voluntarios sanos de 3 países diferentes, Estados Unidos, España y México, y un estudio que duraba 8 semanas. Y eran estudiantes sanos de universidad, estudiantes. Y venían a la primera visita, mi equipo de Miami de psicólogos clínicos les hacía la batería de test, que es la habitual para diagnosticar ansiedad y depresión. Les instalábamos una app en el móvil que iba coleccionando, permitían que nosotros viéramos, ¿no? Obviamente este ya no es el estudio clínico que yo hacía cuando tenía 8 años, aquí habían todos firmado consentimiento y dicho si esto les gustaba o no y qué sensores podíamos ver y cuáles no, todo con HIPAA compliance, todo en diferentes clústeres separados por si había un ciberataque en un lado no nos encontrara en la otra parte de la moneda, o sea, con todas las garantías obviamente de protección del sujeto humano. pues recogiendo los datos de forma pasiva y 1 o 2 preguntas de esos cuestionarios, que son algunos largos, cada 2 días. A mitad del estudio volvían a hacer toda la batería de test y seguíamos recogiendo datos 4 semanas más. Una cosa también importante y que nos interesaba era estudiar en los aspectos culturales y socioeconómicos. Así que el estudio clínico lo hemos hecho tanto en español como en inglés, también para aprovechar que si tienes gente en Puerto Rico, gente en España, gente en México, gente en Miami, que es la ciudad más al norte de Latinoamérica, pues también intentar entender esos condicionantes, ¿no? Así que, bueno, estos son los estudios, los cuestionarios que recolectamos, pues PHQ-9, que es para depresión, GAT-7 para ansiedad, etc. Luego, el aspecto que tiene el app, que es de una empresa existente canadiense, no tengo ningún interés en ellos, espero poder hacer una app aún mejor, pero era lo que tenía, no te dije compro la licencia, pero que podíamos cargar todos nuestros cuestionarios y tener acceso a los 10 sensores que nos interesaban, y aquellas personas que tenían un reloj, pues también usábamos los sensores y los niveles de actividad de ese sensor. Así que ahora, a día de hoy, hemos acabado la recolección de datos, los estamos agregando, tengo estudiantes muy buenos ordenando los datos y agregándolos en características que se puedan comprender, y los están usando para entrenar modelos computacionales que nos permitan estimar con buena precisión ansiedad y depresión. ¿Cuál es lo próximo? El estudio 2, ¿no? Que estamos ya diseñando, no lo tenemos todavía aprobado, va a ser complicado, porque aquí sí que queremos unir, ¿no? Primero queremos tener gente sana, pero también pacientes que tengan depresión y historia de diagnóstico de enfermedad cardiovascular. Empezaremos a enrolar 200 personas y recogeremos no solo los datos pasivos y toda la información de demografía y los cuestionarios clínicos, sino que también los fluidos biológicos, espero que alguno se deje, para poder identificar, ¿no? Ahora intentar aunar la biología con lo digital y poder analizar pues, orina, sudor, sangre y ver si podemos encontrar cuantificar esas moléculas y determinantes de enfermedad de los modelos in vitro. Y de nuevo, agregar todo en características que podemos usar para entrenar modelos y de alguna manera estratificar pacientes, ¿no? En grupos en función de todas esas características y poder tener una manera de monitorizar los pacientes mejor. Y esta es me he ido muy rápido. Ah, no, estoy bien. Esta es la última diapositiva que os quería decir. Es que primero ya el cáncer y otras enfermedades ya lo hemos curado en animalitos pequeños, en ratones ya está curado, porque los cánceres, si son muy pequeños, y es lo que pasa cuando pones un cáncer en un ratón, son del tamaño de un grano de azúcar, un poquito de un grano de azúcar. Yo creo que ha llegado el momento de hacer modelos de humanos, tejidos, órganos, que sean una muy buena herramienta para ganarle la batalla a la enfermedad del humano, ¿no? Yo estoy aquí para curar gente, no para curar un tumor o enfermedad cardiovascular en un ratón. El segundo es que, si os habéis dado cuenta, no trabajo sola y es muy importante romper silos, salir de tu zona de confort. Yo estudié química y me ha tocado hacer biología, polímeros, inteligencia artificial, hablar con psicólogos clínicos. Yo al principio no los entendía mucho porque no entendía el inglés, pero ahora sigo sin entenderlos mucho porque es que venimos de disciplinas tan diferentes que tienes que hacer un esfuerzo muy grande para entenderte, pero que es la única manera de acelerar la investigación, la generación de conocimiento y su traducción a la sociedad. Y el tercero también tiene que ver con el inglés, que yo crecí, ¿no? Y supongo que muchos de vosotros, hay que hablar inglés, hay que hablar inglés, hay que hablar inglés, y sí que es verdad que el inglés sigue siendo la lengua franca, ¿no? En el terreno científico. Os confesaba, ¿no? Que esta charla la doy muchas veces en inglés más que en castellano, pero que para el tema educacional y para impacto clínico cada vez es menos, ¿no? Yo me paso ahora en MIT, pues, casi la mitad de mi tiempo hablando español, ¿no? Porque hay más españoles en Estados Unidos que en España. Y Latinoamérica pues está, ¿no? Hay muchos problemas, mucha riqueza. O sea, para mí trabajar, por ejemplo, ir a Miami es como un parque para un niño, ¿no? O sea, hay tanta diversidad, hay tantos problemas que resolver que es increíble. Y esos problemas vienen en español muchas veces. Y los médicos hablan español. Y bueno, yo creo que solo me falta, ¿no? Esto es uno de mi grupo de alumnos, y agradecer, pues, como os decía, a la doctora Agata La Pedriza, de la Universitat Oberta de Cataluña, a la doctora Marta Pereguerolas, de la Politécnica, y mi mentora todos estos años, va a hacer 25 años que estoy en MIT, a la doctora Martha Gray, y todos el resto de colaboradores, pero sobre todo alumnos muy brillantes que veréis por los nombres, ¿no? Pues son de todo el mundo y vienen y trabajan y luego pues vuelven a sus países y ayudan, ¿no? A crear empresas. Algunos son empresarios, algunos son profesores, y que es muy importante. Y obviamente mi jefe siempre dice no money, no mission. Así que pues tengo que dar gracias a la Caixa, al programa de MIT con México, al programa de MIT con España, Plan Nacional y bueno, a la Fundación Empresas IQS, que es la fundación que apoya a mi universidad, allí en Barcelona, por apoyar todas estas investigaciones y todo este intercambio, que más que nada es mucho intercambio, sobre todo no de conocimiento, que sí, pero de personas, arriba y abajo del Atlántico. Y ya está, aquí ha llegado. ¿Una pregunta?
Muchas gracias, Mercedes. Tenemos tiempo para algunas preguntas. Si alguien tiene alguna pregunta, creo que tenemos a alguien con micrófono, ¿no? Así, ahí hay una pregunta.
Gracias, Mercedes. Si me permites que te llame por tu nombre. Y enhorabuena a la Fundación por organizar estas dos conferencias. La pregunta es, pensando en la curación que has comentado, ¿qué es lo que más puede importarte a la hora de hacer tu trabajo? El armazón que has comentado, ese colágeno, esos fibroblastos, si existen exosomas en esos fibroblastos o estás pensando en ellos, o las células endoteliales. ¿Qué es más vulnerable a la hora de construir ese sistema que tú estás haciendo para que sea derrotado por una molécula o por un mal hábito o por un virus? Gracias.
Gracias por la pregunta. Mi experiencia, cuando estamos en el laboratorio cultivando todos este tipo de células, las más complicadas son las endoteliales. Son esa barrera biológica, ¿no? Que se pensaba que no era una barrera, se pensaba al principio que era simplemente un recubrimiento de los vasos sanguíneos y en cambio realmente hace esa barrera. Y vienen muchos adolescentes al laboratorio y les digo, sobre todo, no abuséis de las drogas ni del alcohol, no hagáis nada, porque claro, a mí me toca ver en el laboratorio lo que pasa con la barrera hematoencefálica cuando le das caña, ¿no? O sea, cuando le pones una droga sintética, droga, quiere decir droga, no medicamento, hablando en español. Pues eso, entonces les digo, no lo hagáis porque a veces también en estos ensayos estamos viendo la reversibilidad del daño, ¿no? Y en algunas de estas el daño es irreversible y eso lo vemos, así que yo creo que las endoteliales son las más difíciles en ese sentido de recuperar cuando hay daño.
Gracias, Merche. Enhorabuena por la charla. Yo te quería preguntar sobre el modelo colaborativo que habéis establecido, muy pionero entre varias universidades o centros de excelencia. ¿Habéis pensado en algún momento en aplicar este desarrollo, este research, al mundo empresarial o al mundo industrial, en el que poder incorporar a alguna gente que no sea académico en lo que es el ejercicio, buscando una aplicación directa de esta tecnología? Por ejemplo, en el ámbito de la seguridad y salud, la empresa maneja personas que trabajan en diferentes plantas productivas, por ejemplo. Bueno, en este sentido, ¿habéis pensado en incorporar quizás este agente?
Gracias. Sí, de hecho yo llevo dos sombreros, ¿no? Hoy me ha tocado representar el de investigadora, pero dirigiendo el programa de MIT con España, por ejemplo, o el programa de innovación, lo que ya está contemplado. O sea, tenemos estudiantes, cada año mandamos 250 estudiantes de MIT a colaborar en empresas de toda España, Portugal y Andorra, y digo España, Portugal y Andorra porque me han añadido países, península ibérica, para no usar demasiado dinero y poder hacer más. Y estos estudiantes pues, vienen a colaborar en empresas españolas, por ejemplo, Acciona, Telefónica, o sea, las grandes, pero también las pequeñas del vivero de empresas de Barcelona, de Madrid, de Sevilla. Así que vienen a colaborar. Claro, en el momento, esto es muy bonito, porque en el momento que tú mandas, ¿no?, a tu hijo a casa de otro, inmediatamente el otro te dice, ahora te mando al mío, ¿no? Entonces esas empresas se acercan a MIT, colaboran a través del programa que lidera Eduardo, por ejemplo, ILP, pero también directamente. Te vienen y dicen, yo he cogido estos alumnos y he entendido, a través de tu hijo académico, he entendido lo que estás haciendo y cómo podríamos esto incorporarlo en la empresa. Entonces, la empresa, hay un mecanismo estándar, igual que hay en España, de colaboración con empresa o con un contrato. Entonces, desarrollamos proyectos y de ahí pues, han salido empresas, como por ejemplo, Aortix, que es una empresa fundada por un exalumno mío que volvió y que ahora está colaborando, que hace parches para tratar la disección aórtica. O sea que hay un flujo totalmente, esto ya se está haciendo, o sea, hay un flujo bidireccional, tanto de estudiantes para un lado y para el otro, como de empresarios y gente de empresa interesada en innovación y en investigación, viniendo a MIT y de MIT y yendo para España. Gracias a, por ejemplo, la Fundación Ramón Areces, que nos trae aquí también. O sea que yo creo que el puente aéreo Barcelona-Madrid existe, ¿vale? Yo ahora ya vengo en AVE, que me gusta mucho más, pero el puente aéreo Boston-Madrid, Boston-Barcelona también existe y es fructífero. Yo creo que siempre se puede hacer más, obviamente.
Mercedes, yo tengo una pregunta.
¿Ah, sí?
Dijiste que en el programa 2 vas a reclutar 200 personas deprimidas. ¿Cómo los vas a reclutar? ¿Vas a tener un diagnóstico de que están deprimidas? ¿O uno dice, yo estoy deprimido y me apunto? ¿Qué tal deprimido tengo que estar para que me tomes?
No, parte del equipo tenemos enfermeras en la plantilla. O sea, en MIT tenemos el Clinical Research Center, que tiene enfermeras certificadas, pero también el equipo de Miami, que son psicólogos clínicos, van a hacer cuando... Nosotros vamos a hacer anuncios, ¿no?, de por favor participa, pero tenemos, dentro de lo que, por ejemplo, tenemos que tener un diagnóstico de depresión. Ese diagnóstico de depresión lo van a hacer los psicólogos clínicos y las enfermeras, haciendo pasar los cuestionarios. También tenemos que decir que no queremos... El diagnóstico se hace con estos cuestionarios y si tienes 25 puntos quiere decir que te estás a punto de suicidar. No vamos a querer gente al borde del suicidio, porque no, pero con diagnóstico de depresión media, media, para ver si podemos, ¿no?, incidir y ver, ¿no?, sobre ese medio que se mejore. Pero sí, no puedes venir y tú decir estoy deprimido. No.
Bueno, hay una pregunta final, la última,
Muchas gracias, Mercedes. Has dicho que estás trabajando en el modelo de tejido cerebral con astrocitos y con capilares. Entonces, para que fuera más extrapolable a la clínica humana, ¿estáis valorando la posibilidad de introducir todas las células que hay y todo el tejido que hay en el cerebro?
Sí, o sea, sí, pero de momento con 2 tipos de células, material y flujo, tenemos suficiente, pero sí, deberíamos de introducir pericitos, deberíamos de introducir neuronas, pero paso a paso, o sea, sí. Pero gracias.
Bueno, yo creo que... Muchas gracias, Mercedes. Ahora tenemos un descanso. 11:45 estamos de vuelta y vamos a continuar con las ponencias del doctor Daniel Anderson, que va a estar muy interesante. Un panel que van a participar él y el profesor Mark Bate, y después vienen los startups. Así que, por favor, les pido que a las 11:45 estén de vuelta y ahora disfruten del descanso. Muchas gracias. Sí. Bueno, retomamos. Muchas gracias nuevamente por venir. Es un placer para mí anunciar al próximo speaker, el doctor Daniel Anderson. El profesor Anderson es un investigador líder en el campo de la nanoterapéutica y los biomateriales. Es profesor en el Departamento de Ingeniería Química del MIT y el Instituto de Ingeniería y Ciencias Médicas, y es miembro del Koch Institute para la Investigación Integrativa del Cáncer del MIT, el Broad Institute de MIT y Harvard, y el Ragon Institute en MGH, MIT y Harvard. La investigación realizada en su laboratorio se centra en el desarrollo de nuevos materiales para la medicina. Ha sido pionero en el desarrollo de biomateriales inteligentes. Su trabajo ha dado lugar a avances en una variedad de áreas, incluyendo dispositivos médicos, terapia celular, administración de fármacos, terapia génica y ciencia de materiales. El doctor Anderson es fundador de varias startups: Living Proof, Olivo Lab, CRISPR Therapeutics, Sigilon Therapeutics, Verso Therapeutics, VasoRx y Orna. Una vez más, haciéndome eco de las palabras de Raimundo Pérez Hernández, quiero aprovechar para felicitar al doctor Anderson por su reciente nombramiento como miembro de la Academia Nacional de Medicina de Estados Unidos. Ser elegido para integrar la Academia es considerado uno de los honores más altos en los campos de medicina y salud, y reconoce a las personas que han demostrado un desempeño profesional y un compromiso de servicio extraordinarios. Ha sido elegido por ser un pionero en las áreas de terapia génica no viral y transferencia celular. Disertará sobre materiales inteligentes para dispositivos médicos y transferencia de células, ARN y editores genómicos. Dr. Anderson, the floor is yours.
Thank you. Terrific. Well, thank you so much for the chance to speak. I'd like to talk a bit about our work on developing what we think of as smart materials for a range of applications, but in particular, so that we can deliver therapeutic cells and genetic medicines in vivo. So I'd like to start off with this slide. This is actually an educational tool developed in the 1980s by Ed Mueller at the FDA. And so if you zoom in, you can see, even back then, all of the different medical devices that people were using at the time. Their synthetic ribs, pacemakers, artificial blood vessels, and the rest. And so, as you might imagine, this is 40 years ago. Today, we have much, much more. But as I think about medical devices, really the first question, if you want to build a medical device, is what material do you use to build it? And so, if we think about the first synthetic materials for medical devices, they were often just using conventional materials that had already existed. For example, vascular grafts. There was a surgeon in Texas who wanted something he could sew with. And so if you're thinking about something you could sew with, you would naturally think of clothing. And so in fact, these first vascular grafts were made of Dacron. And this trend really continued in early medicine. The first artificial heart. So the doctors and scientists wanted something that had a good flex life. Well, what's nice and flexible? In fact, a lady's girdle. And so the first artificial heart was made of the same material as this lady's girdle, polyurethane. And it turned out, actually, that this lady's girdle material did have some of the correct mechanical properties that you might envision for a synthetic heart. But the problem was, biologically, those materials didn't interact well with the body, and specifically in this case, when blood would hit the surface of this material, it would form clots, and then those clots would go to the brains of patients. And so a real revolution in this field has been what we think of as the rational design of biomaterials. So now instead of, when you're building a device, you take an existing material and try to make it work, you actually sit down and think about what are the optimal properties of a material that we use to build a device. And then you go about making that and testing that in a conventional fashion. So one of the earliest examples of a rationally designed biomaterial for a medical device are actually biodegradable sutures. I expect in an audience this size, quite a number of you have had personal experience with these. These are, of course, threads that are used to sew us up after injury and surgery. And because they're biodegradable, when they're exposed to the water in the body, they can break down into simple sugars and be reabsorbed, so you don't need to go back to the doctor to take them out again. Another type of biodegradable suture developed through rational design are shape memory sutures. And so these were developed actually at MIT now over 20 years ago. So these are also biodegradable sutures, but what you can see in these videos is they have shape memory properties that are a function of the temperature. And so when you shift this suture from room temperature to body temperature, it will take on a pre-programmed shape. And on the left, we have a suture that makes a screw. And on the right, you have a suture that will actually tie itself into a knot. So you don't need the doctors. The suture knows how to make its own knots. And to me, these are just nice visual examples of how the rational design of materials is going to continue to lead to important advances. I want to talk about first the use of materials to create living medical devices, and in particular, to create hybrids between synthetic materials and cells so that we can treat a variety of diseases. And so I often start with this picture of Voldemort. We first started work in this area, actually working with the Army, and we're presented with images of quite a few horrendous injuries that people had suffered in combat. And so I choose to use this instead. I think that unfortunately the state-of-the-art therapy, if you have, for example, a missing nose, is that you're going to go to the surgeon and they're going to try to reconstruct what you have left to create a new nose. And if they can't do that, unfortunately then it's sort of the same types of devices you might use for Halloween. So prosthetics that provide some support for the patients but really don't help. But several decades back, scientists realized that it may be possible to actually make synthetic cartilage and tissue using a combination of materials and cells. And here, for example, is an early construct that was an ear made out of synthetic polymers that was then combined with cells that are used to make the cartilage in our bodies. And ultimately, these types of materials can turn into constructs that can be placed in living animals and also in people to replace certain defects. And this is, again, just a nice visual example of this idea that you can combine synthetic materials and living cells to create living therapies for patients that can treat injuries that we previously couldn't treat. And so what I want to focus on in particular is our work on making living medical devices to actually produce drugs in the body. And so the idea is to build a medical device that will give you drugs on demand that are made by these living cells. And so this is a device that is not, for example, a synthetic piece of plastic where you fill it up with drug and then a sensor tells how much drug you want. Instead, these are living cells that actually are paying attention to the nutrients and drugs in your body, and those cells can be programmed to deliver the drugs that you need as needed. And the disease where we've spent the most time has been for the treatment of type 1 diabetes. So most people are familiar with diabetes. This is a disease characterized by loss of control of blood sugar. And in particular, these patients have a condition whereby their own immune system has attacked the insulin-producing cells in their pancreas. So the beta cells are destroyed by their own immune system. And so what we've been trying to do is actually to create a new pancreas for these patients, but one that is actually protected from the immune system. So can we actually combine cells and materials and create something that will sense the amount of blood sugar you have and respond with an appropriate amount of insulin? And the goal is really not just to make a customized device for each patient, but we'd actually like to be able to make literally millions of these things to treat all of the patients that have diabetes in the world today. And so, of course, as an engineer, when you sit down and say, well, I want to build a pancreas, the question is, how do you build it? And what are the design criteria? Well, so as I mentioned, this is a living medical device. And so instead of electricity and drug, you need to supply the device with food and oxygen because these are living cells. In our bodies, of course, those cells are supplied with nutrients and oxygen through the vascular system. Excuse me. But it turns out it's actually a bit complicated to make a new complex three-dimensional vascular system. But it really is essential. In fact, there's an old rule of thumb that if you're building a new tissue that's thicker than around 0.5 millimeter, what happens is that those cells closest to the blood vessel get enough oxygen, but as you move further into the tissue, you have competition for oxygen and nutrients, and the tissue starts to die. And so this is really a fundamental problem. If we want to build a new organ, but we can't get past the supply of oxygen, and we can't build a new blood vessel supply, what do we do? Well, one answer, and this has really motivated most of the field, is just we make it thin. So there's no reason why a new pancreas needs to look like the old one. And this has been work that's now been going on for over 40 years. And so what scientists have been trying to do is actually make little beads that have these insulin-producing cells inside of them. And this is an example of those capsules. So these are small spheres around 0.5 millimeter, and the little pink stuff inside, these are actually pig cells from a pig pancreas that will measure glucose and supply insulin. So in addition to feeding these cells, they need to be able to secrete the key drug, which is insulin in this case. And importantly, they also need to keep out the immune system. And so as an engineer, we think of this as what's called a semi-permeable membrane, where it's a barrier that will allow nutrients and oxygen in, but will keep elements of the immune system out so that those cells will live and function long term. And the reason we need to keep these cells separated is because, especially in type 1 patients, their own immune system would attack and kill islets, even if they were their own islets. You know, and we envision using islets from stem cells that may not be genetically matched to patients. This video is actually an immune cell, in this case what's called a T cell in red, attacking a cancer cell. So this is not attacking a pancreatic cell, but the principle is the same. If we want to keep these new cells alive, we need to physically separate them from the immune system. So how do we do that? Well, it turns out that there is a material in seaweed, not this specific kind of seaweed. Some of you that may like Japanese food probably have seen this before. But there's another type of seaweed whereby you can isolate this sugar, a polymeric sugar, that can be used to create water-rich gels where we can put cells inside. And so what you can do is you can isolate this polymeric sugar, you can put it into a solution and use a conventional droplet generator, and the solution also contains living cells. And then at the end, you end up with these microcapsules that are mostly water, but they have a coating of this polymer and living cells on the inside. And this is not a new idea. In fact, really one of the most important papers in the field was published way back 40 years ago. And what they did is they took this polymer called alginate, combined that with cells, and then asked the question, can we treat diabetic rats with this new synthetic pancreas? And this is actually data from that original paper, which was published in Science back in 1980. And so what you can see here on this axis, this is the blood sugar of the animal, and this is time after implantation of one of two things. In one case, we have cells that are placed in the animal that do not have the spheres, and then we have cells that are in the spheres. And what you can see is that the blood sugar of the animals that received the cells in the spheres actually was lower for a couple weeks. So this was really the first evidence that you could place cells in an animal in a synthetic gel and have those cells measure glucose and secrete insulin as needed to control blood sugar in the animals. But it didn't last forever, and of course the goal is not to have a therapy that lasts 2 weeks, but something that could potentially last the lifetime of the patient. And so as we looked at this early work, the question is, why doesn't it last longer? It actually, these first spheres do everything that we want. They do allow oxygen and nutrients to come in, they allow insulin to come out, they protect it from the immune system. But of course, our bodies and immune systems are smart, and the problem is something called fibrosis. And so it turns out that if you have a synthetic material in your body and your immune system recognizes it but can't eat it, what they'll do is wall it off in a layer of scar tissue. So what happens with these many synthetic materials, in fact most, is that when placed in the body, your immune system will look at it, decide that it's foreign, and say we need to cover this in scar tissue. And so if it's a living medical device, if you have a wall of scar tissue, what that means is that the cells on the inside starve to death. And this is a fundamental problem for many medical devices. In fact, this is an example of a fibrotic scar tissue capsule that was forming around a breast implant. It's a problem for things like pacemakers and eye implants, et cetera. And so, when we also looked at early human trials with these capsules, this is actual data from a patient treated with the first sphere synthetic pancreas. And what you're looking on the left is actually in the abdomen where these were placed. You can see these small spheres embedded in the abdomen. And when you look at them in what's called using histology, which is a form of microscopy, you can see the sphere with dead cells in the middle and then layers and layers of scar tissue. And so our approach to this was really first to ask the question, can we find a better material? One that isn't going to form scar tissue so that we can actually have the cells survive and function. And in particular, what we need to do is identify materials that aren't recognized by the immune system. This is a picture of a macrophage actually eating some bacteria, but it turns out that these cells, these macrophage cells that are present in our bodies, are some of the key early immune cells that recognize these materials and form scar tissue, or at least lead to the scar tissue formation. So the goal was really to identify new materials that could keep cells alive, but sort of be masked from macrophages. And this is a slide for those of you who've worked in biomaterials. I'll go through this quickly, but this is a slide that kind of captures about 10 years of work with 15 people. But basically what we did is we used what are called high throughput approaches to make thousands of new materials. And then we developed systems where we could triage which of those materials is likely to work and which of those isn't. We'd make them using robots. We would make capsules. We would place them into rodents and see whether or not we could see an immune response. And those materials that then looked low, not to stimulate the immune system, we would test in diabetic animals, and ultimately we went on to primates. And so we were able to find a material now that could keep cells alive, but not induce scar tissue formation. And so the key experiment was actually one where we took the pancreas from one primate, we placed those beta-producing cells in these capsules with a new material, place it into a second primate, and then we go back and look and see, does it form scar tissue? And the answer is that it doesn't. And this was really the first time, to our knowledge, that microspheres had been shown to keep cells alive in non-human primates for, in those early studies, 4 months. So we then were happy because we had a new material and a device that could keep the cells alive. But of course, if we're going to treat patients, we need to also have the cells that are going to treat the patients. And when we first started this work, a lot of the field was focused on, in fact, using insulin-producing cells from pigs. Some of you may know that diabetes was first treated with insulin derived from pig pancreas. And in fact, those pig insulin really was an important breakthrough for the field. But we were also very fortunate that around the time we were first developing these new materials and devices, scientists at Harvard had finally figured out how to take human embryonic stem cells and through a series of controlled steps, turn them into insulin-producing beta cells. And it was my colleague Doug Melton, who was at Harvard. He's now a distinguished scientist at Vertex. And I won't step through their process, but what you need to know is in essence, you can create as many human islets as you need in a vat. You can start off with human embryonic stem cells, grow as many as you want, and then over the course of 30 to 40 days, through a series of treatments, you can turn those cells into beta cells that can now secrete insulin. And recently, Doug and the team at Vertex has shown that these cells actually work in people. And so this is Vertex data with their very first patient. And what you're looking at here, it's a little complicated, are 3 graphs. And the graph is basically looking at the blood sugar over during the day for this one patient, first before they were treated with the therapy. And what you'll notice is that there's a lot of movement of the blood sugar into this high level, which is the characteristic diabetic state. Over the course of several months, what they noticed is that the amount of insulin that these patients needed was decreasing. The amount of time that this patient was diabetic was also decreasing. And now, at day 240, the patient no longer needs insulin and is essentially functionally cured. And so this was a really exciting moment for the field. But the fundamental challenge for this therapy is that these cells require immune suppression because these cells do not match the genetics of the patient. And as you might know, one of the challenges with getting a tissue transplant is if it's not genetically matched, your body will recognize it as foreign and kill it. And so this patient has their diabetes treated, but now has to be on immune suppression for the rest of their life in order to maintain this therapy. And so we asked the question, well, can we use these cells that Doug had generated in combination with our devices to treat diabetic animals without immune suppression? And so what we did is we took cells that he created that produced insulin, we placed them through a droplet generator, made our new spheres, placed them into diabetic mice without immune suppression. And this is actually looking at the blood sugar of the animals that were treated over many days. And so for as long as we did this original experiment, we were able to show control of blood sugar in diabetic animals with an immune system with human cells produced from embryonic stem cells. So this gave us hope that this therapy really could be useful in people. We went on to form a company called Sigilon, and Sigilon then formed a partnership with Eli Lilly to work on this in particular for diabetes. And in fact, earlier this year, Lilly acquired Sigilon. So now Sigilon, this company we started, is now a wholly owned subsidiary of Lilly. So, you know, I think we're very excited that it's possible to create living medical devices that could treat diabetes, but we actually think diabetes is one of many diseases where we may be able to, in essence, build a smart reactor that can measure the state of the body and secrete the drugs that you need on demand long term. I'm going to transition now to another challenge we've been working on in the area of biomaterials, which is developing materials to deliver drugs inside of cells. And so it's a fun time for me to give these talks because in the old days, nobody had any idea of what I was talking about. And now most people in the audience actually have had RNA nanoparticles in the form of a COVID vaccine. And so in essence, this is synthetic materials with an RNA delivering this genetic payload inside of your cells to give you an immune response against this disease. And I thought it'd be helpful just to show you the impact that this has had even at one company, which is Pfizer. So this is a chart I found that I show in my class showing the revenue for Pfizer. Then when COVID hit, how much of that revenue came from these RNA nanoparticles. You'll see in 2021 that was $37 billion. And so the point of this slide is not to say that that's the right number, but I think what you can see from this is that the pharmaceutical industry is now paying a lot of attention to these types of therapies because they really are revolutionizing medicine and generating a lot of value for companies working in this space. And so I actually believe this is the tip of the iceberg. This is not just the technology that's going to be useful for vaccines, but in fact, the delivery of nucleic acids, in my opinion, is going to revolutionize medicine over the next couple decades. And the fundamental challenge is not are nucleic acids, things like RNA and DNA, biologically active. The fundamental challenge is can we develop ways to deliver these inside of patients, specifically to certain tissues, in a safe and effective manner. And if we can do that, there are an enormous number of diseases that we might be able to treat. So first, I understand this is a diverse audience, so apologize, but I'm going to give you a little basic biology. If you take biology in college, one of the first things they'll talk about is what's called the central dogma of molecular biology. And so almost everybody has heard of DNA, but the way that DNA works is the DNA inside of our cells is turned into something called messenger RNA, and then that messenger RNA is turned into protein. And it's the proteins that are doing the majority of the work in our cells, whether it's all of the processes, the biological processes of life. And so, as you might expect, If you can deliver messenger RNA inside of a cell, you can, in essence, tell the cell to make any protein that you want. So there's no question that messenger RNA can be turned into protein. And what that means is if you can deliver a messenger RNA into a cell, you can make that cell create any protein you can think of, essentially, with very few exceptions. So just by changing the sequence or the nature of the RNA, you can make a new protein. So this is what we think of as modular pharmacology, the idea that we might be able to make many different drugs to many different diseases with the same basic chemistry. We just tinker with the sequence of the mRNA, and we make a new protein to treat a new disease. So how do we do this? Well, when we conceptualize the challenges of using RNA as medicine, it sounds pretty tough. And so the first step is actually to build a nanoparticle, like we all took for COVID. But for many therapies, we want those nanoparticles to travel through the body to reach organs we're trying to treat. And the way that works is if you inject a nanoparticle in the blood, it needs to travel through the blood, reach the organ of interest. It needs to avoid off-target tissue. It needs to leave the bloodstream and actually enter inside of cells. And so the way that this works is that there's a process of cellular eating called endocytosis, whereby the cells are basically grabbing particles out of the blood and internalizing them. It then needs to escape the cellular stomach. So this is the endosomal escape step. And then once you have a nanoparticle inside what's called the cytoplasm of the cell, it needs to open up and release that RNA so it can now interact with the cellular machinery and make a new protein. And so it sounds like an enormous challenge, but of course we've seen progress. And where we first saw progress were in organs that are natural destinations for nanoparticles. So in fact, if you inject nanoparticles into your bloodstream of many different types of chemistry, they will often end up in some of these organs that filter the blood. So this is places like the liver, the spleen that is a central focus for our immune system. And depending on their size and properties, they can also go to the bone marrow or kidney cells or immune cells. But getting nanoparticles to that organ is not the same thing as getting safe and effective delivery. And this has really been the fundamental challenge for the field. How do we get nanoparticles first to the right organ, but then actually inside those cells in the organ in a way that's working for the patients? And one of the key challenges is that when you think about RNA as a drug, it basically doesn't have any of the properties you'd like to have in a good drug. And so I like to show this picture, which is a diagram representing an example of an mRNA encoding a human protein myoglobin right next to the actual protein that it makes. So if you just think about the size and physical nature of this drug, it really is enormous. And in fact, if you were to put aspirin on there, you'll notice there's a little size bar there that says 5 nm. That means that myoglobin in general is about 5 nanometers, so 1/5 billionth of a meter. If you were to put aspirin up there, it's less than 1 nanometer. So when we compare the challenges of delivering this long, floppy piece of spaghetti relative to a conventional drug, it actually looks quite tough. But of course, we've seen progress. And as I started this talk, one of the first questions is, what are the materials that you use to build a delivery system for a drug like this? Well, it turns out that you can use some of the same materials that are used in our bodies, or at least that are inspired by those same materials in our bodies. So if you look at the surface of a cell, and this is again something that they'll teach you in the first biology class, there's something called a lipid bilayer. So I like to think of this as the skin of our cells contains these compounds called lipids. And what's interesting about lipids is they have a region that likes water, it's called hydrophilic, and a region that doesn't like water, hydrophobic. And so when you place them in an aqueous solution of water, they will basically self-assemble into layers. And under the right conditions, you can actually make nanoparticles out of these lipid materials. And these are some of the structures that you can make in vitro. You can make small nanoparticles, you can make sheets, you can make multi-layer nanoparticles. And in essence, these are the same types of materials that we're using to deliver RNA, both in the vaccines and all the other drugs. And so, for any of you that are chemists in the audience, this is a representation of your typical RNA lipid nanoparticle. So in general, the therapeutic RNA nanoparticles are made of 4 different lipids and nucleic acid. And I won't talk so much about the chemistry of these lipids, but basically what you do is you take a cocktail of these lipids, you take your RNA, and you just do simple controlled mixing, and they sort of spontaneously form nanoparticles on the order of 100 nanometers or less. And these are the kinds of particles we took, as I said, for COVID. So I think where I wanted to go for the rest of the talk is that we all know that RNA nanoparticles can make vaccines, but I think, again, this is really the tip of the iceberg. If you look throughout the field, there's growing evidence that we can make nanoparticles that can go to all of these different organs in the body. In fact, the first FDA-approved drug that was an RNA lipid nanoparticle was not a vaccine. It was a nanoparticle used to treat genetic disease in the liver. It's called Onpattro, and it was FDA-approved in 2018. Most people don't know about this drug because the patient population that it treated was very, very small. But for those of us that have been thinking about nanoparticles as medicine, it was really a seminal moment where in 2018 we could say, look, these aren't just toys that we use to play with in the lab. These are actually things that can treat patients, even if they are for rare diseases. And so I want to give just one example of how we're using these RNA nanoparticles moving forward. And this is actually a question that motivated me to come to MIT. I came to MIT to get more training back in 1999. And the dream was really to make drugs that can actually fix your DNA while you're still using it. And so, really to draw a crude picture to this, say you have a liver disease, which is why I've shown this liver in green. Can you make a nanoparticle that you can inject into the blood, will go to that diseased liver, will actually enter inside of those cells, deliver what's called a genome editor that will then fix your DNA, whereby after repair, now your liver is healthy. And so, again, it sounds like science fiction, but there's now human evidence that these types of therapies are possible. So I'll talk a little bit about that. So the question is, can we build an RNA lipid nanoparticle that can specifically treat genetic disease? And again, to put a graphics to this, what we want to do is find a way to deliver a genome editor inside of our cells so that it can then find its way to the nucleus, fix the DNA, and now the DNA will make corrected copies of the gene in mRNA, and then will make corrected copies of the protein. And so there are many different flavors of genome editing today, but almost all of them involve basically taking advantage of our endogenous DNA repair pathways to create a therapeutic event. So as you might expect, all of us have the ability to repair our DNA to a certain extent. And there are a very interesting collection of different chemistries that the enzymes in our cells are doing to fight off DNA damage. Say if you go spend your day at the beach and you get a lot of UV, there are processes to help you deal with that. But the idea behind genome editing is to create an event whereby your own body's DNA repair pathways will now change your DNA in a therapeutic manner. And so, as I mentioned, there are many different flavors of this. I'm going to talk about one of the simplest and most advanced ones, which is permanently turning genes off through targeted DNA cleavage. And so the idea here is that you can actually permanently turn genes off. So say you have a gene where, for whatever reason, genetically, you're making too much of a protein and that's causing disease. If you can target a double-stranded DNA break, so basically cut it, which is why I've shown the scissors, what happens is that your chromosome opens up, and then there are various processes that will glue it back together again. But if that happens precisely, the scissors will cut it again, and this process continues until there's something called a mutation, either through an insertion or deletion, whereby now it's closed, it doesn't get cut again, but it's caused a defect in that gene that inactivates it. And so the first question if you want to do this, is how do you cut DNA in a correct spot? And, to sort of conceptualize this problem, we have billions of base pairs of DNA. So base pairs may not mean a lot to some of you, but so inside one cell, the calculation is you have enough DNA that if you put it end to end, it'd be about 2 meters. And if you put all of the DNA in your body together, it would actually be the diameter of the solar system times 2. So imagine there is this complex, long sequence, and you want to cut in a precise spot. How do you do that? And in fact, there are now some exciting ways to do it, and one of these is what's called the CRISPR-Cas9 system. And so the Nobel Prize in Chemistry in 2020 was awarded to Emmanuel Charpentier and Jennifer Doudna for their discovery and characterization of the CRISPR systems. And the short version of this story is that in studying how bacteria defend themselves against viruses, they discovered that you can actually create a pair of scissors for DNA, what's called a nuclease, and you can program it specifically to cut, in essence, almost any site that you want. And so this is a molecular diagram of this process. There is this protein derived from bacteria that will create a double-strand DNA break where you want it, and the way you direct it to cut is you use something called a guide RNA. So imagine a protein loaded with a small ribonucleic acid that is programming it to cut DNA precisely where you want it. And so we were interested in, we didn't invent CRISPR, but we were interested in, can we deliver the components of this as a therapy? And if we're going to build a nanoparticle that can do genome editing, what are we going to build it out of? Well, this is a bit of a molecular diagram, but what we realized is that we could deliver this bacterial CRISPR enzyme using messenger RNA. So the same types of nanoparticles we were developing for vaccines and other therapies could be used for genome editing. The only thing that was new is we had to figure out how to deliver this guide RNA. How do you program it to cut in a specific site? And I'm going to spare you a bunch of chemistry, other than to say the focus of our work was really to identify how to make the guide work in a nanoparticle. But after quite a bit of work, we were able to get that to work. And the first model disease we tried to treat was actually something called hypercholesterolemia. So most people are familiar with the fact that if you have your cholesterol too high, it causes heart disease. But what you may not know is there are some patients who have genetic defects that actually lead to very high cholesterol and really enormous injury and morbidity. This is an example of some of the effects that patients can see in their hands due to cholesterol deposition. And typically these patients, depending on their cholesterol levels, suffer heart attacks at a relatively young age. And so the idea was, can we make a nanoparticle that could actually permanently lower your cholesterol? So there are drugs that you can take for the rest of your life to lower cholesterol, but maybe by partially turning off a gene, you might actually be able to permanently lower it. And so we were able to build a nanoparticle that, when combined with a messenger RNA and the right guide RNAs, when given to a mouse, would specifically cut a gene called PCSK9, which is involved in cholesterol. And what we found is, after a single treatment in the mice, 80% of the copies of those genes had been inactivated. So really close to all of those in the liver had been turned off. And when we look at the blood, the PCSK9 is no longer present in the blood. And when we look at cholesterol, it's been lowered by 35%. And mice are a little different, so in humans we expect this to be higher. And so this was a work that showed for the first time you could actually build a CRISPR nanoparticle that can permanently turn a gene off in an animal. And of course, companies have been working to try to get these things to work in people. I'm a founder of CRISPR Therapeutics, and what we've done recently at CRISPR is showed that you can take cells out of a patient, change them genetically, and put them back into patients to treat disease. And so I'm very hopeful that, in fact, the first CRISPR therapies for sickle cell anemia will be approved within a year or so using that approach. But what I've really been interested in is the possibility that you might be able to build a nanoparticle that could correct those genes in the body. And so the disease that's been a focus of this work has been a disease called transthyretin-mediated amyloidosis. Again, a painful name and probably a disease many of you haven't heard of. Basically, this is a disease characterized by misfolding of a protein in the liver that leads to amyloid formation. So many people have heard of these amyloid plaques in Alzheimer's. This is the same problem, but in the liver. And what happens in this TTR disease is those amyloids form in the liver and then travel throughout the body, causing nerve damage and damage to the heart. And so a company called Intellia, to their credit, built an RNA nanoparticle, again using messenger RNA and this chemically modified guide, but now targeting this TTR gene. And this is some of their human data. In fact, the first human data with a CRISPR nanoparticle showing that after a single administration, depending on the dose, you're looking at reduction of this TTR gene activity in people. And so at the highest dose, they're getting 93% silencing. So basically 93% of these copies have been turned off. And so this is, again, a really exciting moment for the field. There's a variety of data that indicate that if you can silence this gene this much, it's likely to be enormously therapeutic in the patients. And so we now have real human proof that these types of nanoparticles can be used to edit the liver. And moving forward, I'm optimistic that nanoparticles will be used to deliver many types of genetic therapy to many different organs, and this is just one example. I thought I would end with a little bit of a story about a new type of RNA. And so there are various types of RNA and DNA that are of interest. It turns out that you can also make RNA into a circle. In fact, if you looked at the RNA in your bodies, you can actually find natural circles in many parts of the body. And what we did was to actually figure out how to make a circle that could produce a protein. And the reason circles can be better than the conventional messenger RNA is because they don't have ends, they can resist nuclease activity. And so this is actually a video from a company I was a founder of called Orna that does a much prettier job of showing circles working. And so basically what we created was a system whereby RNA actually turns itself into a circle, which is really the key discovery. And then we figured out how to program that circle to make lots and lots of protein over time. And so these circles can also be put into nanoparticles. And the first drug that we're thinking of using to treat patients are circle nanoparticles to create CAR T cells in vivo in patients to treat cancer. So what happens is you inject these nanoparticles in the blood, various proteins in the blood attach to the nanoparticle, target it to immune cells, it then will go to an immune cell and it will program that immune cell to attack a tumor. And again, sort of coming back to this video, now in this case, we're actually teaching T cells how to attack cancer using these RNA nanoparticles. I'm hopeful that this will go to patients in the next year or two. And this, like many of these technologies, has generated a lot of interest. So Orna, the company that I founded, has formed a relationship with Merck to work on these circular RNAs for a variety of diseases, in particular vaccines. So I thought I'd end just with a picture of the students and researchers in my group. Actually, this is a picture that's about 9 years old. But at the end of the day, I think people don't appreciate how much of the work is actually done by hardworking graduate students and freshly made PhDs. So with that, I'll thank you and happy to take any questions.
Thank you, Professor Anderson, but I think it would be better if we just move to the round table,
Sure.
to the seats. And Klaus Schleicher will moderate the discussion. Professor Bate, please join us. Así que ahora es el momento para aprovechar para hacer preguntas a cualquiera de ellos. Y será, levanten la mano y Klaus será el que moderará.
So, first of all, thank you for a really incredible presentation. So my mind is right now boggling of all the possibilities of the future. So maybe let me start off with a question that went through my mind on it. So in the next session, we have Catarina Madeira, and we had some startups that talk about their work and the commercialization of some of the research you've done. And you look at the field of biotech, you look at the big pharma companies on it. So what is the role of the startups going to be going forward?
Since I have a microphone and Mark doesn't, I'll start with that. You know, I mean, I think in general, people will tell you that real innovation is difficult to do at big pharma. And in fact, that's why some big pharma companies have been externalizing a lot of their innovative discovery work. I think there continues to be hope that big pharma can sort of reproduce what startups can do, but I mean, they also understand that if you really want to do innovative work, you may need to actually have a venture arm that creates new startups. You may need to invest in academic labs that are actually looking at the problem before it reaches a startup stage. And so I think there's something about an entity that will live or die by a technology that allows it to really push the boundaries of innovation in a way that's difficult to reproduce in big pharma.
What's your take on it, Mark?
Yeah, I think that's very well put. I think as we're seeing with a lot of medicine companies in the startup world, medicines are incredibly difficult to engineer successfully. The comparison I make of engineering a new medicine is really to engineering an airplane. It's one of the most challenging engineering challenges out there. And I think, as Dan said, the innovation that happens in small companies as well as in academia is key to trigger new technology development to get it out into the marketplace. But I think that it is the role of large companies such as Lilly and others that can stick around for decades to actually see these medicines through to the clinic and to patients, because these problems are so hard. And I think we're seeing an evolution in the CRISPR space currently, where a lot of startups that were founded, 5 years ago or so, are facing challenges in the clinic as well. And so I think it'll be interesting to see how that plays out over time, whether there's acquisitions by larger companies like Lilly who can spend the 10+ years that are needed to bring these medicines to patients, or if there's some successes as well. Dan showed some really nice data from Intellia, for example. And then CRISPR, of course, Therapeutics has partnered with Vertex and others to be able to get those to patients. But they're just really, really hard problems, and innovation does have to happen in the startup world.
Right, so I don't know if there's any questions from the audience, I'd be happy to do that. Otherwise, I have a couple more in my head, so there we go.
Hi, my name is Carolina. Thank you very much for your talks. Both of them were amazing. My question is related to how do you see, in your experience, how the regulatory agencies are dealing with these so very much innovative potential therapeutics, especially those that could permanently change, right, the genome for the best, obviously. And many times I feel like innovation goes faster than the regulatory agencies. So perhaps in your experience, you can tell me how you foresee these challenges by the FDA or the EMA, or even by the companies. How are they dealing with these barriers because it's unknown territory? Thank you.
Yeah, I could start. I mean, I think you're right that anytime you have a new technology, there's caution by the regulatory agencies, and I think that's appropriate. I think what we've seen is that with the right data and the right experimental setup, those technologies can advance the people, and we're now starting to see human data, for example, with genome editing, both with nanoparticles and also with the ex vivo therapies, as I mentioned. I'm not a regulatory person, but I think, as you might expect, if you're proposing to permanently change someone's DNA, you need to really be thoughtful about any side effects that could have long term. And I know, for example, that Vertex and CRISPR are planning to follow patients for 15 years after the therapy. Now, if you have a startup and you're thinking about the costs associated with a clinical trial that has to go on forever, you get a little nervous. But I think it's important that we understand how these therapies are going to treat patients over time. And I think the more data that we get, the more comfort there will be at the regulatory agencies. I think the beauty of these so-called modular pharmaceutics is that, in essence, the chemistry is not changing enormously from drug A to drug B to drug C. And so, for example, if you look at Alnylam, which has had a lot of success progressing through the clinic with their constructs, which are modified RNAs, they have an enormous success rate on the order of close to 80% of things that they start get approved. I mean, it's just amazing because the risks are different. Once you start to understand the drug entity, the risks then become more about the disease and the biology. But anyway, I think it's an interesting time, and the more drugs we see progress and get approved, the more comfort there's going to be for these types of technologies.
Yeah, I would second that. I think the CRISPR companies, again, with permanent genome editing that could potentially be in off-target cells is even closer. Okay, that could be in off-target cells is certainly has a very high bar for bringing medicines to patients. I think the antisense oligos and siRNAs and mRNA that's not CRISPR, part of the CRISPR system, has a lower bar because it's a transient medicine on a certain level, like a pill. But I think, you know, the FDA is very cognizant of the need of patients. And so if a patient is suffering a deadly disease, and they need this medicine, then I think, you know, that will be a more permissible solution than, for example, replacing a statin that, you know, 1 billion people take or whatever, right? So I think they adjust the regulatory bar depending on the need of patients and the other solutions that are out there. But the cost is also another huge factor. So Alnylam and others are facing the next sort of generation of cost hurdle, which is as they bring their medicine to very, very broad populations of humans, they need to bring the cost down significantly. And even as siRNAs are relatively inexpensive compared to viral vectors, for example, they're still expensive compared to a statin. Or an antibiotic, much less, right? So there's a combination of sort of economic factors. Are people willing to pay, insurers, patients, and are they willing to take the medicine because of what might be harmful side effects, and those might be very long-lasting. So I think there are a number of challenges that we'll see play out over the coming decade, but again, the innovation in the space is going to drive solutions over time that, as I said in my talk at least, I'm a firm believer that we will pivot towards a nucleic acid medicine generation in the coming century and decades from small molecules alone and antibodies because of the potential they do hold.
Congratulations, Dr. Anderson, for your conference. I am the global coordinator in Madrid Oncology Network. You have told us about briefly about the CAR T cells. My question is, in your lab, in your laboratory, what are the last specific things that you can offer us in oncologic themes, in oncologic aspect, okay?
Yeah, well, thank you. So, you know, it's interesting, the field of nanomedicine in some ways really started with cancer. And for decades, the dream or thinking was that you could build a nanoparticle, inject it in the blood, it would specifically go to cancer cells and it would kill them. But I think what the field has found largely, and some of this might be debatable, but in my opinion, the problem has been that it is possible to make nanoparticles to go to some tumor cells, but you never hit all of them. And so it's really difficult to treat disease that way. What we've seen in the last decade has been a revolution in cancer therapy by actually harnessing the immune system. So not with nanoparticles, but with, for example, checkpoint inhibitors or things like CAR T. And so where I see these types of therapies having an impact in cancer is not by directly attacking the cancer, but by activating or targeting the immune system to go after cancer. And there's a variety of data and different models that suggest you can use nanoparticles in a variety of ways. Just for example, there's human data recently from Moderna and Merck creating an RNA vaccine for cancer. And there are a variety of companies working on creating CAR T cells in vivo in patients, including Orna, or CAR macrophages or CAR NK. And so I personally am optimistic that these therapies are going to affect it not by direct tumor targeting, but by immune modification.
Okay. There's another question back there.
Sí, Mr. Anderson, muchas gracias por su conferencia. Sí, me ha interesado sobre todo las aplicaciones de su tecnología contra los depósitos de sustancia amiloide en el Alzheimer. Pero dado que muchas de sus investigaciones se han radicado en el hígado, pues me interesaría que hablase un poco de esa técnica de reparación.
So I may have missed the translation. I'm going to ask Klaus to ask your question again.
Well, the first part what you said was that the applications of your research was pretty much focused on liver diseases. If this actually can also be applied to Alzheimer in the future.
Sí, bueno, lo del Alzheimer que me ha interesado mucho, pero mi pregunta es si en la cirrosis, dado que muchas de sus experimentos se han basado en la reparación de las células, cuando la enfermedad crónica del hígado se instaura, la cirrosis concretamente, las células están muertas ya. Entonces, la reparación celular sería posible, en teoría, en una etapa anterior, es decir, en la etapa de degeneración grasa. Quizás. ¿Es posible?
I'm sorry, my Spanish.
So my background is not a background, but as much as I understood it, what he just said is that when cellars actually die in the process now, if they can be re-erected, is that correct?
Yeah, well, so let me start first with the Alzheimer's question, because I think there's some complexity here. So one of the dreams of our field has been to create a nanoparticle that can affect the brain. And there have been various studies that have given people hope that it may be possible to create synthetic nanoparticles that can treat the brain. I think in practice, it's been very, very challenging. And the two biggest challenges are, first, when you directly inject into a mouse brain, you can see the majority of that brain being treated. But then when you treat a primate, it's a part of the primate brain. And then if you imagine a human, it's a part of a part. And so it's very difficult to make nanoparticles that can treat the whole brain, even if you apply them directly into the cranial space. I think the real dream has been creating nanoparticles that you can inject into the blood that can cross the blood-brain barrier and affect the brain. And that has been, I think, so far impossible. The only exception is recently there have been clues that certain proteins can be programmed to cross the blood-brain barrier, and they may be able to bring small pieces of RNA. But this is very new and I think still debated by the field. So unfortunately, while there's hope that Alzheimer's may be treated, for example, by intrathecal administration of small RNAs, the broader tool set of nanoparticles, I think, is going to be difficult to apply in the near future. So I don't know if that was the rest of your question, but that was the part.
Once you're delivering what you're trying to deliver, some of the cells are already dead.
Yeah. Can't come back from the dead. I think that's absolutely true. That said, if you really want to get futuristic and dream, there are exciting studies where people reprogram cells to turn them into stem cell states. Can you do some of that reprogramming in vivo in a way that's controlled and doesn't lead to cancer? I can't do that yet. But I think if you were to imagine something that might be able to generate stem cells that could then propagate and treat dead tissue, that might be something to look at.
Okay.
Sí, mi pregunta
Listo.
Vale. Mi pregunta concreta era en el estadio anterior a que las células estén muertas en el hígado, hay un estadio que se llama degeneración grasa. En ese periodo sí que sería posible reparar las células para evitar la muerte celular, que es la cirrosis. ¿Es así?
Right. So the question, as I understand it, was what about diseased liver, that so-called fatty liver that is injured but not dead? I think, you know, the liver has amazing regenerative properties in general, so you can actually cut out a chunk of the liver and see it grow back. Cirrhotic liver, less so. The more advanced, as you know, it becomes a problem. I do think there are opportunities to treat fatty liver with RNA therapies. And I think, you know, it would not be surprising if the toxicity profiles would be different in fatty liver cells relative to healthy normal cells, because depending on the nanoparticle, you may need to give fairly large doses, and so you're requiring the liver cells to survive that therapy. So there may be differences, but I think there is potential to treat liver at that stage. Thank you.
There's a question right over here.
Thank you, Dr. Anderson, for your great talk. I have a question. I don't know if it is trivial, but you mentioned that the nanoparticles are composed mainly of 4 lipids. What is the source of the lipids? Where do they come from?
Yeah, well, I mean, so one of those is cholesterol. So we think of cholesterol as a lipid, and that can come from any sources. But the key lipids are made synthetically, so it's chemistry. But it's not super complex chemistry, honestly. So what we need to do to make these delivery lipids is actually pretty easy compared to what people are already doing to make some of the drugs that we take, the small molecules.
Thank you.
Hi. Okay, thank you for your talks. I just wanted to ask, how are the ethics in the permanent DNA modification? Because I remember the case a few years ago about a Chinese investigator that got punished by the Chinese government for modifying with CRISPR some fetus. And now I just have seen that you have made clinical trials with humans with CRISPR modifications and everything. And I just wanted to know if there is any change in those laws, and also what do you think about permanent changing, because mRNA therapies are good for the health, as we have seen with COVID, but permanent, from my point of view, permanent modification might be like separating groups of people if they can afford the therapies or not because they're permanent. So what are your thoughts?
Start?
Yeah, I mean, I think on the ethical side thus far, I think the demonstration that there isn't genome editing to the gametes, namely the sperm cells and egg cells, the reproductive organs, has been most important. And I think the case you brought up was really targeted germline cell editing. And the ethics around that are, of course, very complicated because you're introducing into the population a permanent evolutionary change, effectively, right, a genetic change that can propagate throughout the species forever. But if you do not allow for off-target editing of gametes, sperm and eggs, then when the patient passes, then the genome editing also passes and does not propagate into the population of humans. So those are two very different application areas. And I think the ethics there are profound, and you know, depending on who you talk to, people will have different opinions, as they always do, in ethical questions. And you know, I don't have one to share, but I think that that's the core issue for the permanent changes that can be introduced genetically that you alluded to.
Yeah, so I agree with Mark. You know, I think the way to think about the work that we and others are doing is we're trying to treat human disease in a way that helps the patient ultimately, because if you can provide a therapy and the patient no longer suffers from the disease and also no longer has to take therapies, it can be transformative. And so I think it's important to look at gene therapy and genome editing the same way you do other drugs. How safe is it, and how efficacious is it, and how does it compare to other ways you can help people? Because some of these diseases, frankly, are awful, and people are going to die if they don't have a therapy, and this may be the only way to treat them. So I agree also about the separation between therapies that do not affect genetic inheritance, like gametes, and therapies that do. I personally feel we should not rule out the possibility that you could do genome editing that would include the gametes so that you know your kids aren't going to have Huntington's, for example. Huntington's is an awful disease. If you could guarantee none of your kids is going to have that, I know I would want to have that therapy. Your second question about cost, I view, you know, I think cost of medical care is a fundamental question and not limited to genome editing. I think it's a fundamental problem for many, many therapies, including basic ones, and it's also a problem we have in the United States. How do you create a functional, equitable system whereby everybody gets the care they need? And unfortunately, I don't have an answer to that one. My only comment is that I think these new therapies need to be thought of in the context of the larger problem of treating patients with drugs and et cetera.
Go ahead.
Hi, it's me again, sorry. So I have a question, because it's directed to both of you, if possible, because both of you have discussed potential delivery methods, nanoparticles in the case of Dr. Anderson, and these nucleic acids, virus-like particles in the case of Dr. Bate. So my question is, we're going to have these different tools, so different delivery methods, and whether there's any preference for the use of one versus the other, including viral vectors, if you want to discuss it, based on preference of tissue or cell type. And in particular, I wanted to raise the case of the bone marrow, whether there's any delivery method that you think could be working better compared to the other one in your experience. Thank you.
Go ahead.
Yeah, so my view on the delivery materials field is that we're at the tip of the iceberg, very, very early prehistoric times in understanding how viruses efficiently traffic through the body. Going back to the CNS question, there are a number of very small messenger RNA viruses that very effectively get into the CNS and transduce cells, but we don't understand even the most basic mechanisms of how that works. There are other viruses, like rabies virus, that enter our neurons in our peripheral nervous system and then retrograde transport through our nervous system to reach our central nervous system. Rabies virus is better understood, but even that virus still has open questions. So the National Institutes of Health in the United States has recently established a very large initiative called the Virome, which is to study viruses from genetics to biochemistry to physiology with a major infusion of investment of capital. And I think that that's going to emerge with understanding that we can later leverage either to make materials that look like viruses, as we're trying to do, or use viruses potentially themselves. But I think what we've learned about viruses over the past 25 years of using adeno-associated virus is that they come with significant challenges surrounding, again, off-target effects, immunogenicity, and cost that have not actually come down despite billions of dollars of investment and thousands of researchers and companies trying to solve those problems. So I do think that the future needs to look to new technologies, but of course I'm biased.
Yeah, I think the only thing I'd add is, we have some tools today to do gene therapy, and we have actually a large collection of nanoparticles and other approaches that have promise. And so today, for example, if you want to treat a genetic disease of the eye, you should use a virus. And Luxturna is an example of an approved therapy. If you wanted to do genome editing of the liver, you would probably focus on a lipid nanoparticle. I know that's what the companies are doing. I think all of these things are going to change over the next 2 decades or so. And at the end of the day, the question is today, what's the best technology for a given application, knowing that it probably is going to be different later.
Yeah, thank you very much for both talks. They've been terrific. I do have a question for Dan Anderson. You've been talking, Dan, about using lipid nanoparticles for drug delivery. And I don't know if you have experience or what's your opinion about biologically derived lipid nanoparticles like exosomes or extracellular vesicles for drug delivery.
Yeah, it's a good question. I think in some ways the answer is similar to the last question, which is that we have the things that are working today, and then we have people working with some interesting data on biological nanoparticles, whether virus-like particles or exosomes or natural lipid vesicles. If I had to place bets, I personally think in the long run, synthetic systems are going to win because they're more easy to define molecularly, more easy to produce and manufacture. But, I'm always surprised by what people come up with, and I wouldn't be surprised if somebody shows me data with a virus-like particle doing some amazing delivery to the brain. And then I would have to change my answer.
So we have about a minute left, and there's a question back there.
Last question.
Thank you for your very exciting presentations. My question is, these small materials are able to deliver compounds that correct not the incorrect gene, but the misfolded protein?
Yeah, so certainly one thing that people are trying to do with genome editing is insert a new protein into the genome. There are different versions. So one is turn a gene off permanently. That's what I talked about. The other is take a genetic defect, say it's a single base pair, and fix it using a base editor. Another is actually inserting a whole new gene into the genome. And there's sort of progressing difficulty with each of those challenges. Today, I think there's a variety of evidence that we will be able to do those things, and the fundamental challenge really is delivery. I think when you think about inserting new genes into the genome, you're now thinking about delivering the genome editing tools, but also delivering a new piece of DNA that's going to be incorporated. I should also add, if you're into all the genome editors, there's something called a prime editor, where you may be able to introduce sequences, some short stretches of DNA without introducing a whole piece of DNA. But anyway, I think there's evidence that we can do these things in cell culture, and there's growing evidence in some animal systems we can do some of these things. But the larger the payloads, the tougher it gets, and the more complex you have with more complexity with multiple payloads, the tougher it gets.
Thank you.
Okay. Thank you, Professor Anderson.
Hang on, I have one more question, which is a personal one. So, being on the frontier now of really gene editing and drug delivery on it, what always bugged me is actually needles. When do you see that we are going to get a wave for drug delivery on needles?
Sorry, could you repeat the last part?
Needles. So right now we have a lot of drug delivery that actually done like vaccines to needles. When do you see the point that we don't need to use needles anymore?
Yeah. Well, I can start with that. You know, I think an example of things that's in clinical trials now are lung therapies where you breathe in a mist of nanoparticles, so no needles. There's a variety of evidence. You can do a vaccine in the nasal cavity, so maybe just a little bit of mist. Depending on what you're trying to treat, you might be able to take a pill. But if you really want to give a large dose of nanoparticle throughout the body, I'm afraid you need a needle.
Yeah. Too bad. Well, thank you very much.
Thank you, Klaus. Thank you, Professors Bate and Anderson. Es un placer ahora invitar a mi colega Catalina Madeira, directora de MIT Startup Exchange. Ella va a presentar a los startups, que es el módulo que viene, pero primero va a introducir lo que es el Startup Exchange para que lo conozcan. Catalina. Ya viene, ¿eh? Ahora viene una parte muy interesante. Son 5 startups tremendamente innovadores, ¿eh? Ok.
Thank you, Eduardo, for the kind introduction. Hi, everyone. It is a pleasure to be here and to present you our program and today's Startup Lightning Talks. Startup Exchange is a program led by MIT Corporate Relations, which is home as well to the Industry Liaison program, or ILP as we call it. The two programs are highly integrated. While ILP primary focus is to take large corporations to MIT and connect them with the institute resources, be it faculty, students, research projects. At Startup Exchange, my team starts by identifying and selecting MIT-connected startups, and then together we connect both parties to help them explore collaborations or partnerships based on common interests and needs. We do this because our mission at Startup Exchange is to support startups grow and expand to ultimately help them solve the world's great challenges. So the companies we support or that are eligible to Startup Exchange, all of them must have a strong connection with the institute, and for that we call them MIT Connected Startups. This connection might come through the founding team, the technology, as well as through their affiliation to other startup programs at MIT, who vet and support the startups themselves. When it comes to origination, at the end of the day, we see startups literally coming from any part of campus. There are over 60 courses related to entrepreneurship that originate many of them, but they can come through any of the 5 schools and 1 college, from centers, labs, as well as from collaborations MIT researchers have ongoing with known MIT organizations. And so this allows us then to onboard companies in our program that are virtually working in any sector. Today, Startup Exchange has a network of about 1,000 startups. They're mostly B2B, and generally speaking, we start working with them when they reach pilot stage. This means that the company is formally created, they have a team that is not student-led, and they have a technology or a product that is ready to be tested or used in a real environment. The exception to this are life sciences startups, as you'll see, because they tend to take much longer to market. On average, every year, we onboard about 150 new companies or new startups in the program. So throughout the year, there's always the opportunity to make new introductions between startups and corporates. Now, within the broad network of our program, there's a group to which we call STEX 25, to whom we provide customized support throughout 1 year. So any of our peers on campus can refer startups to us. We interview them all, and we end up selecting the ones that are a combination of a strong team, solid technology, and a strong alignment with the industry we collaborate with. Each startup is then assigned to a team member who will coordinate the support throughout the year, and this support includes mostly increased visibility and connections to the industry. Then at the end of the 12 months, they leave the group, but they can stay in the program for as long as they are a startup. When it comes to value, startups part of Startup Exchange are exposed to a global network of industry and to its decision makers. And by industry here, I mean mostly ILP corporate members. While the corporates that engage with our program, they are exposed to, they have curated access, to MIT connected startups vetted by our program. Anyone in our network can participate at events. Our office is very prolific when it comes to the organization of events. We organize about 30 conferences and webinars throughout the year. Conferences usually include startup lightning talks like today, and while some events take place on campus, we do design and host many conferences around the globe. And today is an excellent example of that. And this is one of the most valuable offers we have for both sides. For startups, because we bring them to the markets they are exploring, they are aiming to enter, and for corporates, because the events are tailor-made to help them achieve their goals. Our team also launch calls for startups. Many times corporates share with us their internal challenges, or when they're looking for very particular technologies, we launch calls for startups that are featured in our website, and any company in our network can apply to it. A typical next step here is that we facilitate one-on-one meetings between both parties, but these curated meetings can be also requested directly either by the entrepreneurs, part of the Text25 group, or the corporate members. And having said this, I'd like to highlight that our approach is highly customized and curated. We engage with startups and corporates that are all different. They come from different sectors, they have different needs, and what sets us apart is that we don't provide a uniform response to their requests. And so we end up working with them in a myriad of different ways. Our program was created about 8 years ago, and during this time we have Startup Exchange helped ignite many successful collaborations. This slide features a very small sample of those, and I've chosen here the kind of collaborations that we see taking shape more often, such as joint development, even manufacturing, and acquisitions. And now, without further ado, it is my pleasure to kick off the lightning talks. We've invited 5 startups to join us today, all of them working in the health/biotech space. And each startup has 7 minutes to present, and then we'll save the final 3 for a couple of questions. And the first I'd like to call on stage is Dinocardia. Dinocardia is a biomedical device company that developed a new approach to continuously and automatically measure blood pressure. Jose Wong, Chief Product Officer, is here with us today to tell us all about it. Thank you.
Great. Well, thanks, everyone. I'm going to describe a scenario, a problem. We've all had our blood pressure measured before. Maybe you go to the doctor's office, or maybe you have a small machine at home. You sit quietly, you get your blood pressure measured, and what happens when it's high, right? Usually they tell you, relax, we'll take it in a few minutes. You might relax, right? So you take it again, and it's lower, but in fact, what happened was that you changed, but the machine doesn't change, right? So your blood pressure is actually moving. So think about your day. As you go through your day, you have your cup of coffee, you have breakfast, if you have kids, you get your kids ready for school. If you have teenagers, your blood pressure is going up higher. If you go to lunch, and then so on throughout the day till you get to the end of the day, your blood pressure has kind of gone on this roller coaster, right? And time for bed, when you go to sleep, it's supposed to drop, right? That's when you're relaxed. But what we did do was use that first number that we measured at the beginning of the day as your blood pressure, as an indicator of your health, right or wrong. So, but wouldn't it be good if we could actually have better longitudinal data? How big is this problem? So, I'm going to talk about high blood pressure and low blood pressure. High blood pressure is a big issue. It's 1.3 billion people in the world today that have hypertension, right? That's a huge number. In Europe, 1 out of every 5 people is hypertensive. It's a lot higher in the US for lots of reasons. And the majority of people go undiagnosed, which means that they go untreated. It's the #1 cause of stroke and heart attack. Low blood pressure is a big issue in hospitals, right? If you're in for surgery, and we're talking non-cardiac surgery, there's a high risk of post-operative harm. So there's 15 million incidences of AKI, which is acute kidney injury, and then 40 million of myocardial injury. So how do we measure blood pressure today? I think everyone's familiar with the auscultatory. That's where you have a physician and they're trained and they have a stethoscope and a cuff, right? So they're listening for these sounds. They're called carotid cuff sounds. That requires training. You basically get measured once, and then that's your number. The other is this machine, and you've seen lots of them. You get them at the drugstore. You can go to the doctor's office. It's based on oscillometry, which actually it determines a mean arterial pressure, and then it estimates using a ratio your systolic and diastolic pressure. As far as the continuous methods, there are a few out there, but they usually use something called pulse transit time. So they actually look for the delay between an ECG signal and a PPG signal. But those have a lot of issues. They have low signal to noise ratio. They measure capillary blood flow, so skin color and other things are issues. And they also require external calibration. So you're back to putting a cuff on again to figure out what is that baseline number. The only real way to measure continuously today is using an invasive pressure line, which is basically a pressure transducer they put into your radial artery during surgery, and it's a surgical procedure. But we're developing a system that's non-invasive, it's wearable, it's automatic, and it doesn't require external calibration. So if you think about, how would you do this, right? We have to think differently about how we solve this problem. And so we're looking for the tissue response to the artery below. And you can feel this. If you put your fingers on your pulse, what you feel is the force transmitted from the pressure wave in the blood vessel below. So how do we actually capture that information? So we've developed an optomechanical sensor, right? So we use basically a camera and the latest in machine learning and machine vision to look at this area and basically create a force sensor array. We're converting force to pixels, and we use that rich information to reconstruct the pressure wave in the blood vessel below. And from that, we can actually directly say this is systolic and this is diastolic pressure. So our focus is in the critical care. So we're in the operating room and intensive care unit. But as a platform, we see application across a broad range of areas, from remote patient monitoring to consumer wellness. In fact, we're doing studies with pregnant women for preeclampsia and other things, and looking at sleep trials and other things like that. This is an aspiration of our device, and we're getting close to this. We have a prototype that we're using for data collection. It's basically something that's worn on the wrist. It can be wired or wireless. If you're in the OR, you probably want it wired because you never want to run out of power. And then it moves information to a tablet, which gets transmitted to the hospital system. The big thing for us is that it's comfortable, it's unobtrusive, and some feedback we've gotten from some of our users is that they don't even know it's on. Regulatory environment, for the FDA, it's a class 2. For CE, it's a 2B. And we know the requirements to meet, but we're actually looking beyond that because a lot of the standards say you have basically a patient in the hospital and they're not moving much. But we're looking for more real-world data here. Like, so there's motion, people go into the ICU, they're using the remote control, they're eating other things, so they're moving, and a wide range of heart rhythms and patient profiles. So we're conducting all of these trials today. We're actually in the US, and we're looking to expand into Europe and Asia. Really quick, when we talk about real-world stuff, the motion artifact piece is that, imagine we have a unit on one hand and we have the intra-arterial pressure line on the other. We have a movement sensor, which we're able to actually detect the movement. The IAP line, which is the intra-arterial pressure line, actually can't solve for that, but we can, with the exception of a couple beats. This little blip over here that's dampened, that's because there was an occlusion in the arterial line, and that doesn't happen with our device. And lastly, we're looking at predictive measurements, right? So we've done some work recently with a large de-identified data set, and we were able to show with our machine learning model that we could predict a hypotensive event about 10 minutes before it happens. And as far as support areas, we'd love to work with lots of people on strategic partnerships, product development, manufacturing, and clinical studies. We're a very small team, but would love to talk to you more about that. Thank you.
Don't go away.
Oh, no
now
questions.
we have time for maybe 2-3 questions.
So we are planning to have a submission late next year. That's for the FDA.
Thank you very much, Jose. I wanted to ask you, how did you go from the idea stage to actually building the device, and how, it would be like that? Just, did you manage to do the device at university area, or how did it happen?
Yeah, so the original project was one of the postdocs out of MIT. This was out of the computer science and artificial intelligence lab. So there was a prototype that was developed. And it was actually in combination with Tufts University Medical School. So then we started bringing that into more studies, and we've evolved the design, a lot of gray hair, to get to a point where we actually can go out to the 5 or 6 hospitals we're in today to collect data.
Thank you. More of a technical question. I was curious, how do you guys correct for, example, elasticity changes in the skin with age or subcutaneous fat? I would imagine the diversity of the patient population would make it tough to have reproducible analysis.
Yeah, a lot of that has to do with data collection. We think about like the amount of adipose tissue is not as big an issue for us, but you're right. Things like arterial compliance and skin properties. But it also depends on how much pressure is applied in the initial application as well.
Thank you.
Sure.
Pablo Lapuerta, CEO, founder, and director at 4M Therapeutics. 4M is developing targeted therapies for central nervous system disorders such as Alzheimer's.
Muchas gracias. 4M Therapeutics es una compañía que he fundado con la profesora Liwei Tsai del MIT. La plataforma científica que tenemos es basada en investigación de neuronas vivas humanas para identificar medicinas nuevas. Con este trabajo hemos llegado a una oportunidad especial para ayudar a personas que tienen el trastorno bipolar. Tenemos varias ventajas, incluyendo estructuras químicas buenas, un enfoque en el trastorno bipolar que es muy práctico, y estamos muy cerca de un estudio clínico. Esto solo es el comienzo. Tenemos otros productos para tratar al Alzheimer. y para tratar la depresión y otras condiciones. Lo más importante es que tenemos el equipo necesario para hacerlo, con fundadores científicos que son entre los mejores del mundo. El campo de neurociencias empezó con Santiago Ramón y Cajal, que estudió tejidos humanos de personas fallecidas, y con eso pudo presentar la hipótesis de la neurona, que las conexiones entre neuronas podían explicar el aprender. Por más de 100 años, el estudio directo de la neurona humana se basaba en tejidos obtenidos de personas fallecidas, pero ya no. En el 2007 se publicó la habilidad de convertir una célula de la piel a una célula madre. Y esa célula madre se puede cultivar, llegar a ser una neurona. Así que ha abierto un campo totalmente nuevo de crear tejidos cerebrales. Y el líder de este campo es el MIT con la profesora Liaison. También ella está acompañada por el Dr. Haggerty de la Escuela de Medicina de Harvard y Randy Moon, expertos a nivel mundial de este campo nuevo. Ahora, el sistema empieza con estudios de caminos metabólicos que son importantes, basados en la investigación de la doctora Tsai y también del doctor Haggerty. Establecemos pruebas en neuronas humanas de la función de estos caminos y validamos estas pruebas con ejemplos de medicinas buenas que ya conocemos, como el litio. Si ya vemos el efecto del litio en estas células, podemos estudiar muchas más nuevas, a ver si podemos llegar a algo mejor que el litio. El litio es muy bueno, ha ayudado a millones de personas con el trastorno bipolar. El problema es efectos adversos, es un metal y solo puede salir por el cuerpo, por los riñones y daña los riñones. Parte del problema es que damos dosis tan altas que tenemos que medir el nivel de litio en la sangre 5 veces al año. La solución, una medicina nueva que puede dar lo bueno del litio sin lo malo. Un efecto específico que nos ayuda a dar dosis pequeñas y evitar los efectos adversos. Y podemos ver esto. Podemos ver esto en las propiedades de nuestro producto, el 2001. Para inhibir la proteína que el litio tiene que inhibir, pues una concentración muy pequeña. Dura bien, se puede tomar una vez al día, se puede metabolizar, o sea, descomponer naturalmente dentro del cuerpo, evitando los riñones y evitando el daño. Muchos que tienen trastorno bipolar han empezado a tomar estas medicinas que se llaman antipsicóticos atípicos. Son muy buenos, pero a la misma vez causan somnolencia, aumentan el peso y causan diabetes. Lo que preocupa más es que han elevado la tasa de mortalidad en las personas mayores que las toman. Queremos un perfil que evita eso, sin elevar la tasa de mortalidad. Y lo podemos hacer con un producto que se llama el 2001. El 2001 es un producto de metabolismo de ruboxostern. Ruboxostern es un producto que Eli Lilly estudió hace 20 años. Estudió para el tratamiento de problemas del ojo, enfermedades del ojo. Tuvo mucho éxito con un estudio grande, pero falló en otro estudio grande. Y Lilly detuvo su investigación. Unos años después, nuestro profesor Steve Haggerty descubrió que podría ser útil para el trastorno bipolar, pero encontramos que su producto metabólico, o sea, el cuerpo, el hígado, quita un carbón del producto y crea algo mejor, el 2001. Es más potente, más selectivo, más estable. Se puede dar solamente una vez al día y evita interacciones adversas con otras medicinas. Y Cada animal y cada ser humano expuesto a Ruboxostern fue expuesto al 2001. Así que tenemos un perfil que podemos avanzar a estudios clínicos. Es mucho trabajo avanzar. Aquí muestro, nos gustan los efectos de 2001 en neuronas humanas vivas. El litio aquí está reduciendo el nivel de inactivación de una proteína. El 2001 lo hace también, pero lo hace con más eficacidad a una concentración que es la milésima parte de la concentración del litio. También en animales. Estimulamos, pues ves el efecto de un estimulante. El litio lo reduce y aquí el 2001 lo hace más. Estamos ya casi listos para un estudio clínico. Voy a avanzar. Y podemos avanzar también con productos parecidos al Alzheimer's. Ahora, la doctora Sai ha publicado este mayo pues, los efectos de otro producto. Esto es el producto número 4 de nuestro. Y aquí, un ratoncito, lo ponemos en un tanque, nada, busca una plataforma y se queda en la plataforma. Un ratoncito aquí tiene demencia, no puede acordarse dónde está la plataforma, pero con solo 4 semanas de tratamiento, pues ha restaurado la función del cerebro del ratoncito. Es un avance importante. Estamos buscando colaboraciones en el descubrimiento, en ensayos clínicos y en distribución y venta del producto. Y en resumen, tenemos la plataforma, una oportunidad, un trastorno bipolar, unas ventajas importantes y solo es el comienzo. Y tenemos la buena fortuna de tener el equipo para hacerlo. Gracias.
Thank you, Pablo. Who would like to ask a question to Pablo?
Thanks for the talk. Just maybe a clarification. The protein that you were mentioning that we are targeting, the intention is to fully eliminate it, or just to reduce its volume, let's say.
Es que no oí bien. ¿Lo puedes repetir?
Vale, lo puedo decir en español también. Sí. La proteína que mencionaba que se quiere eliminar o reducir, ¿queremos eliminarla completamente o se trata simplemente de reducir su volumen?
Solo reducir su actividad como un 20%, como litio ha hecho en las neuronas humanas que hemos visto. Eso nos ayuda mucho a guiar cuánta inhibición necesitamos. Y no es 100%. Podemos ayudar a mucha gente con solo un 20% así de inhibición.
De acuerdo, gracias.
Any other questions for Pablo? Well, there's one over
Parece
there.
que una aquí.
Enhorabuena por tu charla. La molécula que has presentado, que es mucho más efectiva que el litio, ¿cómo la habéis obtenido? ¿Por screening químico, probando muchas sustancias químicas y ha salido ella como la efectiva? ¿O?
Screening, eso es lo importante de poder crear neuronas, porque el profesor Haggerty ha hecho screening de más de 300.000 moléculas buscando alternativas mejores. Cada molécula estudiada por su efecto en neuronas. Y esta fue una de las mejores.
De las 300.000 probadas.
Sí.
Vale. Muchas gracias.
Okay, thank you. Thank you very much, Pablo.
Thank you.
Next, we'll hear from Leuco. Leuco is developing the first non-invasive white blood cell device. Carlos Castro Gonzalez, co-founder and CEO, is here with us today. Thank you.
Thank you. Good afternoon, everyone. My name is Carlos Castro. I'm the CEO of Leuco. That Oh, now the microphone is working. So I don't know if you heard me. Good afternoon, everyone. My name is Carlos Castro. I'm the CEO and co-founder of Leuco. We are a startup that came out of MIT. Our technology was developed at MIT, and the co-founders came out of that school as well. And we have an innovative medical device that can measure white blood cells completely non-invasively. Now, white blood cells are important for many different patients, but they are particularly important for cancer patients. And every year in Europe and in the US, more than 2 million patients receive chemotherapy. And as a consequence of the treatment, 350,000 of them will require to be hospitalized because they develop infections, while their white blood cells are critically low as a consequence of their treatment. So when these infections and hospital readmissions happen, they bring really negative clinical outcomes, including more than 25,000 deaths, but also more than 10 billion euros in healthcare burden to the healthcare system just coming from the cost of these hospital readmissions alone. And part of why this happens is because current technologies to measure white blood cells, they all require a blood sample. So that means that they can only be performed when the patients go to the clinic, and that can only happen infrequently. And as a consequence, doctors and patients are mostly in the dark in between chemotherapy cycles. So this graph over here represents the journey of a cancer patient on chemo. They receive their treatment on day 1, and at that point they get a blood draw to make sure that their white blood cells are safe enough to receive the treatment. And after that, they go home for 3 weeks approximately, and it is during that time that because of the current limitations from current technologies, there is no way to monitor the patients and figure out which ones are becoming at high risk of a life-threatening infection. So as a result, that only increases the risk of hospital readmission, poor clinical outcomes, chemotherapy discontinuation, and poor quality of life. So this is the critical unmet need that we are addressing with PointCheck. This is our first product in Leuco, and also the first technology that can measure white blood cells completely non-invasively. That means that we don't require a blood sample. Patients just need to insert the finger in our device for 1 minute, and by taking pictures through the skin, we are able to make a measurement that is transmitted to the care team so that they can identify early which patients' white blood cells are becoming critically low, so that they can deploy early intervention that could reduce 50% of these infections and hospital readmissions. This is all based on proprietary technology that we developed at MIT that has been protected so far with a portfolio of 8 patents, and is based on microscopy through the skin of the finger. We particularly focus on that area right on top of the nail, which happens to be a very unique part of our bodies because they are very superficial capillaries that run parallel to the surface. So we can get these videos that you see on the screen. So each of these black loops is a microcapillary, and they are actually so narrow that the white blood cells must squeeze through them one by one. We are using one particular illumination wavelength at which white blood cells are transparent. So if you focus on the narrow parts, you can see some bright spots passing by from time to time, and those are the white blood cells flowing in vivo. So based on this data, we have trained proprietary machine learning algorithms that identify and count those cells and are able to make a measurement. We have done extensive clinical validation in collaboration with hospitals in Spain, including Hospital La Paz and 12 de Octubre. Also hospitals in the US. We have worked with Mass General Hospital and MD Anderson. And so far we have tested this on more than 190 patients where we are showing excellent diagnostic accuracy compared to the blood draws. That accuracy is exceeding the requirements that FDA and CE Mark are expecting for approval. And we have published this in peer-reviewed and top-tier journals. So with this validated product, we have access to a sizable market because of the hospital readmissions that we can avoid. The total savings total 3 billion euros in Europe, and similarly in the US. Those are our initial markets because that's where we currently have collaborations, but this could be a worldwide product. And in those markets, we have a B2B business model. Our value proposition is that we can reduce the cost of those hospital readmissions by 50%. We can also help monitor the side effects of drugs during clinical trials. Our beachhead customers are healthcare providers and insurers, as well as pharmaceutical companies. And we have already demonstrated a lot of traction, including 2 commercial pilots with pharmaceutical companies, and letters of support from leading cancer centers that are willing to adopt our technology as soon as we get regulatory approval. So in our timeline, we started in 2019 when we raised a seed round of financing. With those resources, we were able to develop the core technology and complete our customer discovery. And those milestones help us raise a Series A late last year. And the goal of this stage of development is now completing the initial pharmaceutical pilots, as well as getting to the CE mark and FDA approval, completing our clinical trials for regulatory approval. So at that point, we expect to do another round of financing so that we can launch the product into the market. And beyond that, our vision is to expand this to other patient populations and applications beyond cancer, but also develop the technology so that we can not only measure white blood cells, but also other parameters in the blood, or completely non-invasively. This is the team behind LUCO. We are 4 scientists and entrepreneurs that we met at MIT. The team is composed by scientists, engineers, medical doctors, and entrepreneurs. We have a lot of experience producing top-tier science, developing relationships with strategic partners, and getting awards at business plan competitions. And we complement this team with an experienced board of advisors that includes key opinion leaders in oncology, scientific experts, and also consultants that can help us with the regulatory and biostatistical aspects of the project. We currently have ongoing collaborations in Spain. We work with the Technical University of Madrid for the core development of the technology, also with Hospital 12 de Octubre for the clinical validation. With Telefónica, we are currently developing commercial pilots as well as integration with the 5G network. And we work with Fundación Madrid +D for the translation of the technology. We are currently looking for new collaborations, including partnerships with cancer clinics, healthcare insurers, pharmaceutical companies, and medical device distributors. So we are LUCO, the first medical device for non-invasive white blood cell monitoring. With this technology, we have a unique opportunity to radically improve the clinical outcomes for millions of cancer patients and other immunocompromised populations. So if you want to join us in this mission, please feel free to contact me, and I will be happy to take your questions now. Thank you.
Thank you. Thank you, Carlos. Any questions for Carlos about LUCO?
Congratulations for your talk. Very interesting. Can you distinguish which type of white blood cell are you looking at? At least between lymphocytes and granulocytes, or?
Yeah, so that's a great question. Actually, for cancer patients in particularly, what the oncologists worry about is neutrophils. That's the most common type of white blood cell, the one that fights infection. So actually, our algorithm, all of the white blood cell subtypes, they look very similar in our optical system. But there has been a lot of literature that describe features that are specific to neutrophils. They are slightly larger than other subtypes. They also are brighter because they produce more backscattering. So we have tuned our algorithms so that they can pick up white blood cells that comply with features that would be characteristic of neutrophils. So we are maximizing the likelihood of detecting neutrophils.
Mm-hmm.
Then we don't know for sure because we don't have the ground truth for every cell that passes. We don't know the subtype. But then when we compare to the reference, to the gold standard, to the blood draw, we get that high diagnostic accuracy. So at the moment, we are tuning the algorithms to detect neutrophils in particular.
Mm. Thank you.
Right. Thanks. So just a quick question, not technical, just about the use case. You mentioned that the idea is to be for the companies to be a B2B service. So if I am a patient going through a treatment, the way it would work is that the hospital would give me the device. I would have to take it at home and use it as prescribed. So there would be no additional costs to me as a patient. I wouldn't have to buy it or rent it. Is that the idea?
That's the idea. This was based on a lot of customer interviews. So cancer patients, they had limiting willingness to pay out of pocket. And also our value proposition is to patients because we can improve their clinical outcomes, but also to the healthcare insurers because they bear the cost of these hospital readmissions, each of which in the US is a $46,000 event. So the idea is that this technology would be covered by the healthcare insurers. That's the value proposition because they can save a lot of money. They can also offer better treatment to their patients. So once you are diagnosed with cancer and chemotherapy has been determined to be your course of treatment, your oncologist would prescribe our solution, and that way you would get the monitoring during the course of your chemotherapy.
In one of your last slides, you mentioned future applications. What is the team considering?
We have two axes of future applications. One is for this initial product that can measure white blood cells. We can apply it to other patient populations beyond chemotherapy. So there are more than 10 million patients in the US that suffer from other immunocompromised conditions, and they could benefit from increased monitoring of their white blood cells. And that includes multiple sclerosis patients, organ transplants, autoimmune disorders. And we could access all of those populations with our initial product. So that's one axis of growth for us. The other is developing the platform technologies, capabilities, I guess. So right now we train our algorithms to detect white blood cells, but we have this unique opportunity to image the blood non-invasively. So we think that we can train it to detect other parameters in the blood, including hemoglobin, hematocrit, sickle cell. So those are future applications of the technology as well.
Okay, cool. Thank you.
Thank you.
Next, we'll learn a little bit more about Cache DNA. We have Heather Williams with us. She's the Vice President of Clinical Genomics Operations and Chief of Staff at Cache, that is improving molecular preservation.
Yeah, okay, I have it already. Yeah. Good afternoon, everyone. As you might remember from earlier today, we're here because of work that was started in Dr. Mark Bate's laboratory by our co-founder, Dr. James Banal. I just want to say that I wouldn't be up here upstage without this lovely team that we've assembled who have expertise across chemistry, genomics, automation, and computational biology tools. We're a small but mighty team. So I am a clinical laboratory geneticist by training, and I get very excited about genomics. And I guess lucky for me, genomics is everywhere in the news, from sequencing newborns as soon as they are born, looking at genetic screening across large populations, and even applications in the arts to look at maybe why Beethoven was deaf. And I'm even excited when it's in the news and it's completely wrong, but don't worry, I'm here, I fixed it. It should be mutation. So right now we're experiencing this explosion in minimally invasive medicine. There was a question earlier about when are we going to transition from no needles. That's not today, but we're getting there slowly but surely. So some of these laboratory platforms that are using a simple blood draw to access the information in your genes are things like non-invasive prenatal screening that's given to pregnant mothers, and liquid biopsy cancer screening that's used for both predisposition to develop a cancer and also to monitor disease status in patients who've been previously diagnosed. So everyone is super excited about genetics, just like I am, but how do they manage this behind the scenes? Well, you saw this earlier today. This is really messy. There's these freezer farms. Someone's very proud to show that they don't know where anything is in their freezer, and we need to solve this. Again, you saw this earlier. Everyone wants to keep their samples. It's too expensive to keep them now, even if you don't have even if you have the ability to sequence them. And of course, everyone's doing it differently based on those images you just saw. So what is Cache doing here? So we are a simple way to store and organize biological samples, and the current ways that everyone is doing that is obviously inefficient. So you've heard from many of the speakers today about how we have growing healthcare challenges from diabetes, cancer, Alzheimer's, but all of those diseases are being led by genomics-informed care. And then, of course, we have challenges with accessing these samples. All the ways in which samples are in a fridge or a freezer, it's disorganized, and you can't give patients the cutting-edge sequencing technologies in that kind of landscape. But obviously, we're storing these samples, and the biobanking market is expanding. And one of the major issues we actually have with these stored samples is that over 80% of genetic studies that are performed to date are studies of samples from primarily only European populations. And that's a big issue as the diversity of our global population changes. And of course, people are trying to address the diversity issue, but there are extensive costs associated with that. So what is our technology? Again, you saw this earlier. We are using the basis of biology to protect nucleic acids in an encapsulant, and that allows us to store it free of a fridge or a freezer, so at ambient temperature on a bench somewhere indefinitely. How do we know that this works? Well, our company was a part of the Illumina for Startups. You might be familiar that Illumina is about 80 to 90% of the market in sequencing platforms, so it was great to be a part of that organization. And the way that we know that when you put something in our encapsulant to protect it from the environment is we performed studies and evaluated a sample that was in a freezer and a sample in our technology, and we have over 95% concordance across clinically derived DNA samples preserved over time. And this is very important because if a technology or freezing were to introduce a change, clinicians and laboratorians could not interpret what that genome means. So we have a variety of different applications of this technology. Every life on this planet has DNA and RNA, but I'm going to focus on oncology because that's primarily where I've spent the last couple of decades in patient care. So this is a diagram of an ongoing study that we have with a large cancer center in North America. This cancer center is accredited to an international standard that is also used here in Spain and across Europe. And essentially what we're doing is we're working with them to evaluate cache as the pause button for these samples. So you might recognize that when someone has a cancer diagnosis, and in this image, breast cancer, that tumor is excised, it's eventually sliced, a pathologist will review that morphology, and increasingly those classifications of disease for prognosis, diagnosis, and therapy are dependent upon molecular signatures that exist in the DNA. But here in Spain and across Europe and the world, we cannot access these tests because of costs and other contributing factors. So often these samples will be biobanked, and as they sit in a freezer or a fridge over time, they actually degrade. But enter cache, we can pause it. That sample will be the same as when you put it into our technology as when you want to take it out. And of course, that means that you can use that unaltered DNA, interpret the information, and add to the literature about cancer, about your patient status, obviously increasing the ability of us to store samples indefinitely and ultimately access that information. So we are seeking partnership, and we have ongoing partnerships with academic medical centers, diagnostic laboratories, and biobanks, and commercial partners and life sciences tools companies that are interested in amassing biobanks of samples or analyzing genetic information from a variety of different clinical specimens. I'd be happy to talk to any of you about that, and thank you so much.
Thank you, Heather. Who has questions for Heather about cache?
All right, thanks for the talk. I'm afraid that this might be a stupid question, but I'll let it out nonetheless. So I think it was Professor Baith, sorry if I mispronounce, who mentioned that DNA, right, which is what we try to store, is a very stable molecule by itself, and we find it in fossils. For example, in mammoth was an example, without any kind of storage facilities. Let's say, why then do we need these solutions if that's?
Yeah, I would say DNA in comparison to RNA is quite stable. But one of the things that we know, I think it was a woolly mammoth was in the image, we know that those DNA samples, although we can retrieve them, there are lots of issues with retrieving them. It's low quality DNA. We need a lot more sample in order to draw conclusions. And when you're talking about clinical care, sometimes you can only get a small portion of tumor or something of this sort. And when you put it in a freezer or a fridge, it actually decreases the fidelity of that sample over time. I didn't show an image, but there's a very famous one where you put a sample such as DNA in a freezer, and at 10 years, there are multiple changes from environmental pressure, consequences of crystallization, all of that. And we want to just get around all of that. So stability, yes, but there are other things that happen to DNA over time.
So by which way you help in keeping the stability of the DNA? How?
Yeah, so we take the extracted nucleic acid, just like everybody does in the clinic, and then instead of going straight to sequencing or straight to a freezer, we add some reagents. It creates the encapsulant, and then that can be stored on your bench. And when you want to de-encapsulate or have the nascent nucleic acid back, it's a 10-minute procedure. It's ready to go. You can do library preparation and go all the way through the clinical workflow.
Mm-hmm. Thank you.
Maybe I'm confused, but they did say something about, like coming up with exabyte USB sticks around this concept. Do you have anything can you tell us anything about this?
Okay. So I think you're referring to Dr. Bate mentioning that DNA can be used as a hardware to encode. So I would say right now we are focused on the storage of these biomolecules, but you can obviously envision a future where we have amassed these samples. They can be stored indefinitely at room temperature, and then we can leverage that data in a way that's informative for research, discovery, pharmaceuticals, things of that sort. I think he also had on the slide, many other companies are working on that, so I would say stay tuned for that aspect of the technology.
I have a closing question for you. How will using this technology decrease the cost of genetic tests for patients?
Okay. So I mentioned in my talk about many oncologists and genetic counselors who are offering these tests across Europe are saying, well, it's great that we have these, but only the major centers have them. There are insurance issues and things of that sort. So if you can imagine in a regulated lab, you have to pay for freezers, you have to pay for 24-hour staff. Everything has to be watched because this is patient care, right? And when we transition that to a room temperature technology, all of those costs go down. And so then you can imagine instead of billing thousands of dollars for a test, you can cut that down using our technology. So essentially that increases the access to these tests because the cost is reduced.
Okay. Thank you, Heather.
Yeah.
Now, to wrap up the startup lightning talks, I'd like to call on stage Inés Herrero, founder of Biomixing. Biomixing has developed new agitation systems for biofermentation reactors.
Thank you. It works. Hello. Before starting, I would like to ask you, do you know what have in common? The yogurt I have this morning, the face cream I'm wearing, COVID-19 vaccines, many biotech products or recent cancer treatments? Yes. Yes. Well, they are all produced inside bioreactors. So many people don't know what bioreactors are, but they are really everywhere. Yes, so they are used, yes, in pharma, they are used in cosmetics, they are used to produce culture meat, and they are used also in traditional food manufacturing and in many fermentation processes. It's a huge market, and we focus mostly on pharma and biotech food. There are many needs in this sector. They are very expensive. Also, the processing times can be very long, and we can remember this from when we were fighting for COVID-19 vaccines, for example. The quality sometimes is not good enough inside the bioreactors because sometimes they are large. Inside, you need to treat the culture as if they were in nature. So you need to mix all these microorganisms or cells so that they are mixed with oxygen, with nutrients, and you need to mix them well. But if you don't mix them well, you may have some homogeneity problems or shear stress. You can have problems with that. Energy consumption can also be very high because some of these bioreactors can be huge. We worked for to designing some bioreactors for Repsol, for example, and they were huge, as huge as a house. And there are also many steps in the scaling processes. What do we propose? We have developed an innovative agitation and ideation system that can be used either to retrofit existing bioreactors or it can be included in new bioreactors. What can they do? Well, we have designed them using numerical simulations based on complex mathematical models and also on our knowledge of fluid mechanics. With this, we can create like a digital twin that allowed us to know how it performs, how the culture behaves inside the bioreactors. The better design of the agitation and ideation system allows us to have energy savings of up to 40%, or to have a higher quality product, or to reduce the mixing time by up to 75%. So at the end, we help our clients to save money on time. How do we do it? What is the technology behind? Well, out of all the components that are that compose a bioreactor, we focus on the two that impact more on the efficiency of the bioreactor, which is the agitation and ideation system. The design is based on fluid mechanics and fluid dynamic principles. And sometimes we have asked our clients, this is your new bioreactor, and they say, yeah, that's the old one. The new one has a new software, it may have a new control system, but or new materials, right, single use, but the inside, the agitation and ideation system, with very few exceptions, they have remained the same for more than 30 years or even 40. This is a very simple example. We have one of our clients' bioreactor on the left, and on the right is the same after we retrofitted it. You can see that the bubbles are much thinner, and they are much more homogeneously distributed inside the bioreactor. This is just an example with water and air and in a glass vessel so that it could be seen. But for this client, we decrease the processing time by 50% and with less shear. Oops, sorry. These are just some of the clients we have had, different types of bioreactors, different sizes. Well, this is also another example. In this case, what they wanted to reduce was shear stress, which was reduced from 0.80 nearly to 0.6. How do we do it? We normally study our client's bioreactor, doing the simulation, proposing alternatives, or what we want to achieve, and that's where we are now. We would like not just to create customized products, but also to get to some standard products. So we would like to offer like 4 or 5 types of agitation and ideation system for different groups of cultures, right? So we could do that for clients working with standard cultures. And well, we would like at some point to get in touch with bioreactor manufacturers so that we could offer them our biomixing inside system. What are we looking for? Well, we look for collaborations with proof of concepts. We would like to collaborate also with academia so that we can help our clients to more sustainable manufacturing, because we can reduce energy consumption when working at the industrial scale, and also to make bioprocesses more efficient, like reducing processing times or increasing quality in different sectors, right? I think that's it. Thank you. I forgot our awards. Anyway, never mind.
Thank you, Ines. Any questions about biomixing? Yes.
I can see a large market for your product. What do consumers need to buy from you? Is it just for propeller, or do they need to install a new bioreactor?
No, it can be just the propeller, I mean the agitation, and depending on the case, also the ideation system. Yeah. I mean, in some cases with the agitation system is enough. In some other cases, the ideation is really so inefficient that we also change that. Mm-hmm.
Okay.
Go ahead.
Okay, thank you. I was just trying to understand, so the new part is the design of the propeller itself, because the principle is the same. You're still using a propeller ride together with aeration. So what's the new part? Is it the way it's the design process? From what I've seen, you're using a simulation, so it's just an improvement of an existing design, basically. Is that it?
I mean, it's the agitation, the propellers, that they have a specific geometry that improves substantially the mixing. And what we have patented is not just the device itself, but the process, right? So well, everything is protected, the whole system. Mm-hmm.
In one of the slides, you showed us different demonstrations you conducted so far. To which capacity were you able to scale?
We are focused in fluid mechanics. So for us, the size is not the problem. We have worked with very, very tiny bioreactors, but we have also designed bioreactors as large as a house. So the key is how the liquid and the fluids move inside. Yeah, and some people ask, well, why other people don't do the same, or where is your innovation? Why other people don't do the same? But really, to design it, it may look simple, but it's very complex because you need to know lots of complex mathematical models. You need to know aerodynamics. You need to know fluid mechanics. That reduced quite a lot the number of experts. Those experts normally are working in Airbus or in Boeing, but they are not in biotech companies. Mm-hmm.
Yeah, so, oh yeah, okay, it's working. So about the product itself, clarification more than a question. So it has to be custom made for each customer, because I assume that all those properties that you seek to improve, for example, like the shear, depends on the type of fluid that you're working with. So for each individual customer, let's say you have to adapt the design. Mm-hmm. That's it. It's not one product.
It is both, right? So up to now, we have worked very, very much offering a very, very customized products, right? Product. But we can standardize it. In fact, bioreactor manufacturers currently, they offer 2-3 types of impellers, right? So we could offer, in fact, we are talking with some bioreactor manufacturers, we can offer like a range of another 4 types of agitation and ideation systems, right? So that they could be, each of these types could be specific for a group of cultures, right? Because inside the bioreactors, some cultures really behave similarly in terms of oxygen demand or shear sensitive, or so we can standardize it. quite a lot. But still, there are many companies that have very much customized bioreactors. So for them, we would have to adapt it to their bioreactor.
Thank you, Ines. Thank you, everyone, for your questions.
Thank you.
Okay, so we're about to close this symposium. I would like to express my gratitude to the MIT faculty, connected startups, and ILP colleagues for making the time to come here to Madrid with their in their busy agendas. Quiero agradecer también muy especialmente a la Fundación Ramón Areces por ser siempre tan buenos amigos y socios en esto. Es siempre un placer venir aquí a Madrid y espero verlos en la próxima conferencia. I would also like to say thanks to the remote attendees for joining us today. Gracias por todo y espero que termine bien el día.